Good morning, madam. Hello. Good morning. Sonal, madam. Good morning. Hello. Am I audible? Hello.
Yes, sir, you are audible.
Yeah. Aji, madam. Good morning. Uh, Good morning, Dr. Sonal.
Sir, good morning,
Good
sir.
morning. You just announce one time because people there might be knowing 11 o'clock we are starting program. So you can just announce the participant they are already joined to contact their friends and ask them to join first. Okay.
Yes, sir, we are sending messages in the WhatsApp groups.
Yeah.
Yes.
Because some of the participant in the mind that it's 11 o'clock.
Sir, we have started giving the messages yesterday itself. Let's see. We can wait for
Okay.
5 minutes.
Yeah.
Yes.
Speakers join, I think. Selukar. Selukar is there. Hello, good morning. Dr. Selukar is
No, sir,
not
I
yet joined.
think he's not yet joined.
Just call him. Call him. Join.
Yes, sir. Yes.
Good morning, Dr. Selokar.
Hello, sir. Good morning.
Good morning. Good morning.
I'm audible, sir.
Yes, yes, you are audible. Dr. Sonal.
Hello, sir. Shall we start?
Yeah, because more than 108 participants have joined. I
Okay,
think
sir.
we can start.
Rest will keep joining, sir.
Yeah.
So, again, a very good morning and a warm welcome to all the participants, our speakers, the chairperson, co-chairman, and my team from NAHEP PAFSU. Today, friends, this is the last technical session for our training on gene cloning, advances and applications in veterinary sciences. We have 3 lectures for today, as we had already instructed. The first presenter or the first lecture will be, the first speaker will be Dr. Naresh Selokar. We are honored to have him. He is working as a scientist in Embryo Biotechnology Laboratory at Animal Biotechnology Center, ICAR, NDRI, Karnal. Dr. Naresh Selokar, who has completed his BVSc from Nagpur Veterinary College and Masters and PhD in Animal Biotechnology from ICAR, NDRI. He was also a postdoctorate fellow at FLI Institute of Farm Animal Genetics, Gene Editing at Germany. Dr. Selokar has 12 awards and recognitions at his credit. Also, he has around 40 publications in PubMed and with more than 537 citations. Now, his current research areas and specialization basically, it includes the production of cloned farm animals, the CRISPR editing techniques in farm animals, IVF in cows and buffaloes. He's also trying to understand the molecular mechanisms of genome reprogramming process in cloned embryos and stem cells. His team is also working on molecular reproduction. Friends, we are honored and we are very proud to have one of the key members of the team of scientists from India who produce the cloned buffaloes. So definitely Dr. Selokar will be sharing his experiences of animal cloning and how they could manage to prepare buffalo clones, their centers. And I think his PhD thesis was also based on this kind of study. This technical session will be again further conducted by the chairperson, Dr. S.B. Maji ma'am, Professor of Department of Veterinary Microbiology at Mumbai Veterinary College. And the co-chairman accompanying her will be Dr. V.S. Vaskar sir, Professor of Department of Veterinary Public Health, Kranti C. Nana Patil College of Veterinary Science, Shirwa. Friends, Dr. Selokar will be talking about gene editing applications in farm animals. I request the chairperson, Dr. Maji ma'am, and the co-chairman, Dr. Vaskar sir, please carry over the session now. Ma'am?
Good. Am I audible, Sonal?
Yes, ma'am. Yes.
Yes, madam.
Yeah. Good morning to all, and welcome to this session on the final day of this NAPcast training program. I am delighted to welcome Dr. Selokar for this morning session. So without wasting much time and having such a great biodata, Dr. Selokar, I invite you to please start your talk and enlighten us with the topics. Thank you.
Hello. I am audible.
Yes, yes, sir. Yes.
Okay. A very warm good morning to all participants who are attending this 1 week international training on the gene cloning and advances and applications in veterinary science. First of all, I am grateful and thankful to the organizing team, Dr. Ingole, Dr. Vilasar, and Dr. Sonal, for inviting me and giving me the opportunity to share what we have been doing, particularly in terms of the research area, that is the gene editing, which I've been working from last 2 or 3 years. I also congratulate to the team for the organizing this 1 week of the training, particularly highlighting the importance of the biotechnology that is a gene cloning. I think in today's time, in today's world, we have been so many challenges. One of the challenge for us to provide the food to the human being, obviously the human being, as well as to protect the health of the human being. Not only, but now that today we have the one health concept. So to protect the health of the animals also and protect the welfare of animal also, because in the pandemic time, the COVID has been taught us the many things, is that the human is not an alone. If we have been talking to the protect the human, we need to also protect the environment, protect the animals, their production, their health. So in this direction, I must congratulate to the team that they have been organizing this, the gene cloning or molecular biology techniques, or the kind of the work we have been doing in the not only in the country but worldwide. So the topic has been that I'm today will talk for about in our time is about the gene editing. I think this is a very recent technique or very recent research has been done, but not only in the animals, but in the more majority of the work has been done in the crop science, particularly to improve the production potential of the crop yields as well as to make them a disease resistance. Because we seen that our aim was to, like for a cow, we have been aiming to produce a more milk. In addition to that, we also protect them from the avoid the any kind of the infection or the disease, for example, FMD or the brucella or the any genetic disease. One of them is the now the current pandemic COVID. Anyway, so I will just start sharing. This is the slides are visible. My slides are visible. Hello, I'm audible. Slides are seen,
Yes,
visible.
you are audible.
Yes, Dr. Naresh.
Okay,
So,
We can see
yes,
your
we
slide.
can see it.
okay. So the topic gene editing and its application in farm animal, I will, because the as I know that there are the 60 or 70% people are there from the student, either the masters or PhD. So I will try to highlight some of the basics of the gene editing. I think from the last the speakers, couple of from the last 4 or 5 days, the people have been talked about the gene. That is a gene cloning itself as the different types of these cloning methods. One is a gene cloning, one is the animal cloning. There is a gene editing, reproductive cloning, therapeutic cloning. There's a different kind. So we will go one by one because the I hope that there are the students. So I will just been try the basic principle of the gene editing, and then I will go to the applied aspects, and then we'll shift to the kind of the research work we're doing at the NDRI. Uh, so what are the gene editing techniques? What are the tools are available? If there are the gene editing, I think everybody, I hope that many of the people, they have heard about the CRISPR, that is a designer animals and crops, or even people have been talking about the designer human being. But there are the before this in evolution of the CRISPR technique in 2014 or 2013 when the first publication came, and I think the last year the Nobel Prize has been awarded to the two ladies who have been worked on the CRISPR, Jennifer Doudna and Sharpinter. The Sharpinter was the postdoctoral fellow in the Doudna lab, and they have been used this CRISPR technology to exploit or to manipulate the genome in the stem cell as well as the mammalian cell. But anyway, before the CRISPR, there are other tools are available. One is the restriction enzymes. I think restriction enzyme, any biotechnological lab or for any gene cloning, anything with the restriction enzyme is used. The restriction enzyme is a kind of the enzyme, they have been cut on the specific target sites in the DNA. They have specific nucleotide sequence they recognize. And this restriction has been many years from the decade has been used to make and specific changes into the genome, or if you wanted to delete something or some wanted to change anything in the genome. The second one, the zinc finger nucleases and the challenge gene editing, these are the protein based chemistry, but the CRISPR is the is based upon the nucleotide chemistry. So we will talk a little bit about what is a zinc finger, talon, and CRISPR. I will more focus on the CRISPR based editing. Why the CRISPR based editing is more, because first of all, it's an easy to use in any laboratory. So it's very small infrastructures are required. If you have the refrigerated centrifuge or kind of the basic facility, multiple laboratory easily can be used. But particularly for the zinc finger nucleases or the talon editing, it requires the protein laboratory. Protein means you need to have the well expertized laboratory of the proteomics, and then it can be used in the laboratory. So anyway, what these all the gene editing tools has been do, they enable to make the changes in the DNA. And if the changes in DNA means either deletion of the DNA, addition of the DNA, or the replacement. For example, if we are talking about, suppose in the case of the gene cloning, we insert a target, suppose PCR product into the vector for the sequencing. That is one of the, and for that the restriction enzymes has been used. But suppose we're talking about the produce, the animals, which has been showing the desirable phenotype, or for example, the secretion of the insulin into the milk of the cow. In that case, the human insulin gene need to be placed into the genome of the cow, and that need to be directed toward the mammary gland so that it will produce into the milk. So through this, particularly the CRISPR editing of the talon and zinc finger, it is possible that any gene into the genome is from the tiny ants to the gigantic elephant can be modified. And the people has been attempted this in almost all species, and the crops has been very various kinds of the varieties of the crops. Also, the model domestic animal has been produced. So, but I will talk more about the domestic animal. Now, at what place the CRISPR, because the CRISPR is itself stands for a cluster regularly interspaced short palindromic repeats. If you talk about the globally, if it's still in India, and this is a data from 2018, still India is in 15th place position. The majority of the research work has been done on the CRISPR technique is either in the United States or in the China. And majority, if we talk about the agricultural application, it includes the crop science as well as the animal science or species science. Still the more majority of agriculture application is done by the China, but major of the publication or citation has been done is basically in the United States. But anyway, what we need, because this is a potential application of this technology for any biological science, either it is in biomedical science, agricultural science, or industrial science, where the biology has been applied, we need to focus what we actually need, because in India still the very few people they have been working on the gene editing technique. So what we need to invest more. We need to have to provide the funding to the people as well as to more scientific strength. So we need to train to the people, educate them to the people so that they can use this technology in their laboratory. Now why we need to think about the advanced techniques, for example, gene editing or gene cloning or advances, the biotech techniques. I think everybody with agrees with me is that India is going to be the most population country soon. We will going to surpass the China. It's expected that we surpass in 2060 like that, but 2050. But I hope that the growth of the Indian population, it will be surpassed, and we will be the more populous country. So what the challenge we have? The challenge we have is that we need to provide food to the people, as well as we need to protect the health. I think this pandemic has been teachers. Not only we have any kind of extra medical infrastructure or any kind of the facilities we have, but still we need to have been challenges of the health issues. For example, this is a COVID situation. So to have this situation, we need to use this all the advanced techniques. So to protect the health as well as to provide them the food. I think in 2050 we are expecting country or the world expecting the 10 billion human population, and that to provide them to, we need to use these all the kind of the technique, but with the caution, with that there should not be hampering to the environment, to the hampering to the threat to the future, like this COVID. Now we will talk of the detail because there are the students. I will go slowly to the what the CRISPR actually stands for. The CRISPR, it is the different components. The CRISPR is a crRNA. This CRISPR RNA, it contains the guide RNA. I think DNA, everybody knows DNA is a double helix. It has the nucleotide ATGC, and these nucleotide, they have been coding to the protein. One amino acid coded by 3 nucleotide, and this ultimately determine the any message or any phenotype into the animal or the plant or anything biological system. So what how the CRISPR has been recognized? The CRISPR as the one component, the CRISPR RNA, another component, trans-activating RNA. I think this has been, if you look at here, this is the green color mark here. This is a crRNA. This is a CRISPR RNA. What CRISPR RNA actually do? It recognize the 20 nucleotide sequence into the genome. If this 20 nucleotide sequence will recognize, and in combination with the trans-activating RNA, it's called the trans-activating RNA. This is an important part, though it is not recognized any DNA sequence into the genome, but is an essential to activate. There is a protein that is a Cas9 endonucleases. So endonucleases is no endonuclease is a kind of, it is maybe the you can say is a restriction enzyme which cut the DNA into the specific site, and that is impossible to cut the DNA. But what the endonuclease Cas9 is, the endonuclease is a protein or the kind of the restriction enzyme that, along with the crRNA and the transactive RNA, recognize this the 20 nucleotide stretch, and they have been cut into the third and fourth position of the DNA, and they make the double stranded break. Double stranded break means, I think you know, the DNA is a double helix. There is a two strand. So the Cas9 protein cut both the strands, and when the nick is there happen. So what happened when the nick is there? Then there will be the repair pathway initiated. One is the non-homologous end joining repair pathway. Another is the homology directed repair pathway. But anyway, the important also is noting that there is a PAM site, there is a protaspace adjacent motif site is there, which is that the NGG. So N is a any nucleotide, GG is a glycine. This nucleotide, this is the nucleotide required to recognize the 20 nucleotide, and this 20 nucleotide they actually determine which site we are targeting. For example, we are targeting to knocking out of the beta lactoglobulin gene and not to express into mammary gland. So we need to designing the CRISPR. This is a crRNA against the beta lactoglobulin gene only, and then when we transfect or deliver this CRISPR components into the cells or embryos, then they will perform the editing, and only the beta lactoglobulin gene will be the knocking out. Similarly, if you wanted to insert, for example, human insulin gene into the mammary gland, then we need to provide the donor template. Donor template means the stretch of the nucleotide, that is stretch of the nucleotide for the coding the human insulin, and that need to be inserted into the mammary gland, any mammary gland protein promoter, for example, beta casein or lactoglobulin. This is the basic components of these CRISPR, which has been used. So how CRISPR has been discovered, the person Francis Mozica, when he was doing as a postdoctoral fellowship, postdoctoral work at the Spain, he has invented that CRISPR. CRISPR is not the new technique is like the 2014, but earlier it was been majority you work in the bacteria. So bacterial immunity, the CRISPR has been playing a very important role. So then later in 2013 or 14 onward, it has been used for the mammalian gene editing. So how in bacteria, when the bacteria is infected with the virus, we call it the bacteriophage, there is a DNA strand is there or their genetic material. In the genetic material, this is a spacer sequence. What happens is in bacterial genome, there is a repeat that is called the CRISPR array. Cluster means it is an array. Regularly means every sequence interspace. There is interspace region. Interspace short palindromic. I know I think the student of the biotechnology or molecular biology or even microbiology, they know the palindromic means what? It is a madam. If you read left to right or right to left, is a meaning is same. The same nucleotide. Cluster regular into short palindromic repeats is there like 1 2 3 4. So what happened when the bacterial the fast DNA inserted into the spacer sequence, that happened that recognized as a memory of the bacterial chromosome, that genome, bacterial genome. What happened in the next time when the infection happened, when the Cas9 complexes and they have been transcribed into the gRNA, they form the active Cas9 complex. And the Cas9 complex, when the second time of infection of the same bacteriophage or same viral, bacteriophage means the virus of the bacteria, that time they recognize, because this is the same sequence has been coming from the previous infection, so they will cut the genomic DNA of the bacteriophage, and this way bacteria protect them as an immunity. And this bacterial principal immunity system later been exploited for the mammalian genome. And I think everybody knows that in 2020 in the chemistry, these two ladies, the Sharpinter and Daduna, has been awarded the Nobel Prize for developing the method of genome editing, particularly for the mammalian as well as the plant cells. Now, hello.
दोन्ही
Okay.
कानात टाकले तरी चालतील पण त्या कानात इन्फेक्शन नाहीये याच कानात इन्फेक्शन आहे 200 रुपये
Okay, what are the working principle? I think if you will talk about the gene editing, particularly for the animals, we need to work on the embryos. There is a zygote which has initiated the life. So once we inject this,
कधी
the
झाली की मग इंजेक्शन आहे ते देऊन टाकू
they will target loci is there, and then it will cut the CRISPR editing will cut the double stranded break. And as I inform you that there are two pathways. One is a non-homologous end joining pathway, another homology directed pathway. So indel formation, indel means insertion and deletion. It will lead to disruption of the function of a target gene. For example, knocking out of the beta lactoglobulin. Similarly, suppose the homology directed pathway will activate when we provide the donor template. So if you provide the donor template means, for example, the human insulin gene need to be expressed into the mammary gland. In this case, this donor template will be human insulin, and this need to be targeted into, because our target site is the beta lactoglobulin promoter example. So it will be inserted into the target site. When the animals lactate, that time it will secrete, because the promoter has been driven by the mammary gland protein. So this way it can be, this is a basic working principle, is a straightforward working principle, and this can be exploited in any even in the now the people have been trying to knocking out of the gene in bacteria to see the pathogenicity. Or so suppose there is any tuberculosis bacteria, so bacterial genome need to be edited, which gene is actually responsible for the pathogenicity. So not only in bacteria, in animals also, sorry, not only in animals or these stem cells or the somatic cell, but it has been also been exploited in the bacteria, yeast, fungi, and in all the kind of these microorganisms or the plants and the crops. This technique has been exploring. Now, what are the forms of the CRISPR? This is just I will overview, because some of the students may have been informed that is the expression plasmids are there. Is a plasmid. I think this is a gene cloning. There's an important part is a plasmid is very important, because if you wanted to clone any gene, you require the plasmid to multiply them, to transform them, to characterize them. You need to require the plasmid, and then clone it, transform it, and then sequence it. So plasmid bed chemistry is also be available. Active proteins also been available. mRNA sequence can be available, are available, and the viral particle, adeno-associated virus. And these all the form of the CRISPRs can be used for the genome editing. Each one have their different advantages and the disadvantages. Once we have been in our laboratory, we have either plasmid-based chemistry or the mRNA or the ribonucleoprotein. Then they need to be delivered into the cells. For example, the mammalian cell, or for example cow cells. So there is a different methods of delivery is there, the viral vector, lipid nanoparticle, and the electroporation. I think I will not go into the detail about the how the viral vectors or lipid nanoparticle or the electroporation. I think maybe some of will have been already taught when we have been transfecting any kind of the gene into the any kind of the vectors or the mRNAs or the siRNAs or the gene editing component into the cell. So gene editing, then after the delivering into the cell, this function, they will function, they recognize, and they do the genome editing. Now, this is the efficiency. We talked about the efficiency of the different the proteins. There is a plasmid base, mRNA base, and the protein. Obviously, protein has the highest efficiency, because in mammalian cell, if you transfect or deliver either the DNA, there's a plasmid, or the mRNA, it will need to be transcribed into the active protein. If mRNA is there, it need to be translated to become active protein. And in case of here, so if you talk about the efficiency, the plasmid have the lower efficiency, and the mRNA has a medium or moderate, but the protein, because that is immediately and ready to use. If we talked about the cost, obviously the protein is the most costlier to generate the protein, or to purchase, even the purchase protein is a costlier. But to have the plasmid, because very simple, that's why Even though the low efficiency, this plasmid-based chemistry works well in many type of the experiment. For example, in somatic cell, because somatic cells is large in number, so they have been available. mRNA is moderate again. If you talk about the specificity, how specifically they have been recognized and cut or do the genome editing, though plasmid has the least specific as compared to the protein and the mRNA chemistry, they are the more specific. Now, coming into the CRISPR, what are the CRISPRs been exploited, or how the CRISPR has been exploited? The CRISPR has been exploited to produce the crops or edit the crops and animal. For example, double muscle mass. Our laboratory at NDRI, we have been working on the production of double muscle mass buffalo to increase the meat yield, or allergen-free peanuts is one some of the example. New also some of particularly for the human diseases, hemoglobinopathies. For example, sickle cell anemia. I think we know that sickle cell anemia, it is a point mutation, is one nucleotide change in the hemoglobin has been lead to the formation of the disease for the sickle cell anemia. So people are trying to replace that one amino acid with the correct amino acid. I think valine has been need to be replaced with the glycine in case of the sickle cell anemia, so that there should not be any deformities in the RBC, and that need to be recovered. This is under the phase trial or clinical trials. People has already been corrected that one, but it's not been approved yet. Also, there is a strategy to provide the antibiotic and antiviral resistance. I think antibiotic and antiviral resistance is in, I think, big issues in the in animals as well as in the humans, that we have been becoming resistant to the antibiotic. Also, in China, he has been he is a scientist from the China. He has been attempted to create a designer baby by knocking out of this gene responsible for the AIDS, that is HIV. So they wanted to make a baby such that they are not infected with HIV. But all the legal issues, I will talk about my next talk, how the legally or regulatory issues has been covered, particularly about the gene editing technique. But in human, there has been completely banned to have any kind of the research, particularly for the germline editing. Means you cannot change the germline. That has been completely banned in human in all over the world. We'll talk about in the next session about these regulations. If you talk about the CRISPR research, it has been going to increasing by day. If you look at here, that these the many people, they have been coming, many laboratories now been exploring this the CRISPR editing technique for their research. Either they are working on the plant or bacteria or animals or human trials or human clinical trials, or the anything. The people has been increasing. This is all about the basic what we I would like to share, particularly for the student. This is basic. Now coming to the part which which we have been dealing in India and our laboratory. So as my profession as a veterinarian, and among is a reproductive biotechnologist, my aim, what if we talk about the Indian situation, what is the two major foods, particularly from animal source? Either we require the milk, we rear the animals for the milk or for the meat. So if you look at here, though we have been from the last many decades, India has been forefront in the milk production. Despite that, we have been ranked first in the milk production. Our productivity of animal are the less. So what we have been trying to improve the productivity of the animal, not the production. Productivity means each day how much milk needs to be produced. Similarly, if you look at how the Indian meat, how the Indian requirement of the meat, particularly of the poultry, sheep, and goat. So that need to be think, because still there's more than the 70-80% of Indian population, they are the non-vegetarian. So from the many years, how does we improving the yield of the farm animal, either in the milk or meat? I think selective breeding we have been doing. So selective breeding, we know that we have developed the many new breeds of the cattle. For example, India have developed the current Frise or current Swiss. These are the breeds or crossbreed has been developed. Similarly, now we have been talking about the genomic selection in farm animals. In addition to that, next what we have been trying to improve the nutritional interventions. So there is many nutritional interventions has been done to improve the productivity. And now the new technologies we are talking about, the biotechnological tools. One of them is the assisted reproduction, that is an OPU IVF. I think MAPSU also working on the OPU and IVF at Nagpur. Similarly, we are talking about the gene cloning or the transgenesis, or all the biotechnological tools, so that we can improve the productivity of the animals. Now, why we eat why we need to edit, I think I will not do. Improve the trait, animal human health, and the animal welfare. So how to edit the farm animal? If you look at the classical breeding, hello, okay. In the classical breeding, in the favorable, unfavorable animal, this is we have been doing. This is a classical breeding example. In form to improve any upgrade to the animal, it requires 8 to 10 generations, and that is a part of the classical breeding. But with the help of new gene editing techniques, for example, gene finger nucleases, talent, and CRISPR-Cas, within the one generation, because we know that which gene are responsible for the particular trait, and that because of that we are targeting into the gene and improve the productivity or achieve the favorable animal population within the one generation. And that is in terms of the editing in the sperms and the oocyte. This in zygote need to be produced, and in the zygote stage only we need to have the all the kind of the editing. Some of the important application of the genome editing in domestic animals. The in the United States, the hornless cattle has been produced. This hornless because hornless cattle, their aim was to protect the not only the caretaker who has been handling to the animals, because we also doing in the debudding in the India, but it is a manual debudding. But they have the genetically or genome editing tools can be used to make the cattle hornless. Again, the resistance, again, the bovine tuberculosis. I think the tuberculosis is one of the important disease. That is a genetic disease that India has been talking that we wanted to eradicate the tuberculosis from the India by 2030. But I don't know, because ultimately, unless and until we target the bovine tuberculosis or buffalo or bovine tuberculosis, it's very difficult to eradicate the tuberculosis from the developing countries, at least from the developing countries like the India. So in China, they have been produced these increased resistance against bovine tuberculosis, in which they have been target the gene. It's an NRAMP. This is a because I think the tuberculosis, some of people may know, the tuberculosis intracytoplasmic bacteria which has been proliferate, localized, and from the colonization in the macrophages. So this is the gene in the macrophages immunity. So their aim was to increase the macrophage immunity, and then so that the animals are resistant to the tuberculosis. Similarly, this animal, the gene, they are CD163. They have been used in the United States and University of Edinburgh, UK. They produce the bovine reproductive and respiratory syndrome virus infection, particularly for the pig, because in their country, the pig is an important the meat animal. So these gene has been edited, and this now been the scientists from their respective countries, they have been talking to use this particular knocking out animal or this edited animal for the breeding, so that they not become too infected with the disease. Another one, in case of the ruminants, for example, cow, buffalo, sheep, and goat, the myostatin is a negative regulator of growth, and if you're knocking out this myostatin gene, then there will be increased muscle mass. Similarly, in pigs, there is a nanos2 gene has been ablated, so there is no formation of the oocytes and the sperms in the pig. This study has been done in the pig. So what I would like to highlight is that through the gene editing techniques, it is quite possible to manipulate any gene and to achieve the improved productivity. For example, the muscle yield here with the myostatin, or to make them animal disease resistant. For example, resistance against the bovine tuberculosis. Now the progress I will come into the kind of work we have been doing in our laboratory. So progress what has been done in our laboratory, not only our laboratory, but because recently I have been joined to the NDRI. Previously, I was working at the Central Institute of Research on Buffalo. So I will also highlight what you have been done in NDRI as well as at the CIRB and our new initiatives on the genometric research in farm animals, both in the buffaloes, sheep, and goats. Now, before that, if you wanted to produce any kind of the genome edited or transgenic animal, we need to have a well-established assisted reproduction protocol. So what we have been in NDRI, we have been well established or developed the technique called the buffalo cloning technique. So just I will highlight how the buffalo clones are produced. So if you wanted to clone any buffalo or any animal, we need to have these somatic cells from their animal. The cloning is different than the IVF. In IVF, both the sperms and oocytes are required. But in case of the cloning, there is absolute no requirement of the sperms. As well as the IVF technique, the genome and the offspring has been coming 50% from the male side, 50% from the female side. But in case of the cloning, that 100% genome is coming from the animal whose somatic cell has been used as a nuclear donor anyway. So what happened in to produce the clone embryos, we need to have the somatic cells in the laboratory. Similarly, we are regularly blocking the slaughterhouse ovaries. From the ovaries, we isolate these oocyte, immature oocyte, and these oocyte need to be mature in the laboratory for 21 hours after maturation. There is a process to make them enucleated. The nucleus need to be removed from the oocyte, and what the technique we develop at NDRI, in which the two enucleated oocyte is electrofused with the one somatic. If you look at here, this is a small dot in between the two enucleated oocyte. It has been electrofused. Once they are electrically fused, these embryos need to be activated and culture in the laboratory for the 7 to 8 days. After 7 to 8 days, the developed embryo, there's a different stages of embryo from the 2 cell, 4 cell, 8 cell, and 16 cell stage. Once they reach to the blastocyst stage, they are transferred into the surrogate animal, surrogate or the foster mother. So this animal is need not to be means inseminated. Once the animals come into the heat, they need to be left for these embryo transfer, and embryo transfer on the seventh day, this seventh or eighth day, the embryos need to be transplanted to the surrogate mother. Once this is established in the pregnancies, then there is then after, in case of buffalo, three three 110 days plus minus 10 days, so 320 days around, the calf was born, and that calf is the genetically identical to the animals from where we use the somatic cell. So this animal, the replica of this animal has been produced through this cloning method, and this cloning technique is well established in our country. So what are the clones currently available in the country? So at NDRI, it has been working from 2005 onwards, and has been produced the several clones in the NDRI has been initiated. When at the CRB also, they have been working on the buffalo cloning in 2013 or 14 onwards, and they have also now been working on buffalo cloning. So at the current, we have around the uh, 20 clone buffaloes. They are live. We have produced more than that, but some of the clones has been died. But at presently, we have the 20 clone buffaloes, of which the 16s are the bulls. The 16 clones are the bull, and these bulls can be used, can produce the 2 million semen doses for the breeding. So what we are currently trying to do, we have been done these kind of the studies in these clones, that they have been produced. Once they have been born, they mature well, suppose they attain the puberty, there is absolutely no abnormalities, and they are donating the semen. If you look at here, in case of females, some of the females, they have delivered 4 or 5 calves, so they have the normally reproduction is there, normal production is there. Similarly, in case of the female, in case of the male, the some of the bulls, this one, this is a bull produced at the CRB in 2015. This bull has been now producing semen and more than 25 progenies. Progenies means through the use of the artificial insemination, and this indicating that those clones are available, they are maturing well, they are being can be used for the breeding run. This is our the next target, to convince to the government or convince to the agencies of ICR or DADH that we have the clones, and if you have wanted to use them for the breeding purpose, because these all the clones we have been selected at from the elite animal, elite bulls. This for the breeding program has been done in the buffalo, and then these are the clones of either males or females. They are from the elite category. So another, this is the just an highlight that at the CIRB, there are the more than the this is the 7 clones are produced from the single animal. The single animal, single bull is there. They have been cloned to 7 clone. Also reclone bulls of these earlier clone bull named the Gaurav. So these have also been one of the achievement in buffalo cloning. Similarly, we also clone the Assamese buffalo. This sample has been brought from the Kanapada Veterinary College, Assam, Guwahati, and then produce the clone at the in Haryana. So it indicated that from any part the somatic cells can be collected, because somatic cell collection or culture is very simple steps. We need to have animal. There is no injury to the animal, just the tissue, small tissue biopsy like the blood collection. It will bring to the our laboratory, and then it will be used to produce the clone animal. This we also been proved. Similarly, we also been trying to produce the clone animal from the somatic cell isolate from the frozen semen. This animal has been died, But if you wanted to restore them for the genetics, particularly suppose any bull is there, it's a very famous bull, somehow it's the animal has been died. If we have the semen is available in the biobank, semen biobank, there is very semen biobank, or the attempt has been made to establish somatic cell varieties of them, then any time this kind of the animal can be restored, either male or female, if their cells are available in the frozen state. As I was talking about that, we have been analyzing the fertility of these clone bulls. So analyzing of fertility of clone bulls, that we have currently compared the three bulls, more bulls we are going to compare. So we analyze the fertility, IVF based fertility and AI based fertility. That I was talking that one clone bull has more than the 25 clone progenies, and this is indicated that the clone bulls, if you look at here, the clones Resta, Swarna, Nisargaro, and this is a non-clone bull, they are absolutely have the similar fertility in case of the IVF as well as in case of the AI-based fertility, they are similarly fertile. Now coming into the transgenic work, we have been established the protocol how the transgenic embryos. If you look at here, these are the transgenic green embryos. Is there? If you green means is a GFP reporter protein to the Venus, the reporter. This reporter gene. So in this, what we have been done, we electroporated. Electroporation means that you're giving the electric shock to the cell and transfer the vector, that is a plasmid, into the somatic cell. Once they have been integrated into the genome, that need to be proliferated. Single cell colonies has been done, and they generate these clonal selection. That is for the cell cloning is there. So from generation of the more population of cell from the single cell, that technique is called the cell cloning. So this way we produce these uh, and this cells, and then after this cells are produced, these are used as a donor cell to produce the embryo. That is a clone. If we look at here, this is a handmade clone embryo. They are Juna free. That's why these embryos looks like different. But this is a blastocyst. This is a transgenic blastocyst. They are expressing these venous proteins. So this protocol we have been established. We also established the frozen repository, and this repository currently available at the CIRB, my previous institution. If anybody wanted to interest work on the somatic cell sample, so they can request to the director CRB to have the assets of the biological sample, particularly frozen somatic cells of the buffalo. And pedigree of the buffalo, these animals are the elite in nature, so every information will be provided. And at presently at the CRB, majority of the somatic cells are either from the Murrah breed and the Nili Ravi. These are the two important breeds of the buffalo. Now coming into the our recent the project which I've been working. This is an NSF funded project. This project is a NDRI is a lead center. In addition to that, the CIRB, Hisar, CIRG, the Goat Institute from the Makhdoom, and Coast Kashmir. Though we are coming together and we initiated this project to produce the double muscle mass farm animal using the CRISPR. This is our target. To next couple of years, we wanted to produce the genome edited animals, particularly for the improved meat yield. So in these, the sheep, goats, and buffalo, these animals is selected the myostatin gene. I was talking that if you're knocking out the myostatin gene, then there will be the improved in the meat production. So what we'll do, our aim was to edit in the somatic cells or embryos using the CRISPR technique. Once when we do the editing, we need to do the couple of assays to check that this editing has been proper. For example, T7E assays or Sanger sequencing or whole genome sequencing. This need to be done once we have been edited cells in our laboratory. So these edited cells will be used to produce the handmade cloning, because this cloning technique which I have been talking from last couple of minutes, to just highlighting that if you wanted to produce the transgenic animal or gene edited animal, your laboratory must have the excellent infrastructure or the workforce or the manpower for this, either the cloning or IVF or the micromanipulation. This technique, the person need to have expertise in this technique. Without that, is really impossible to produce the transgenic or genome edited animals. So the somatic cells will be used to produce the edited animals, as well as in embryos. If you electroporate or micro injected the RNP complex and to produce, after this embryos will produce, they will transfer surgically in case, non-surgically in case of the buffalo, and surgically embryo transfer will be done in case of the sheep and goat. And following that, the animal need to be produced. This is a model animals which including the improved meat yield. The improved meat yield, so we need to check, because in the CRISPR there is a one important point or consideration need to be off target. Off target means sometimes what happen, because the 20 nucleotide stretch is many, some of the sites they also have the similar nucleotide sequence. The CRISPR unanimously cut that sites and because of that there is off target. Off target means other than the target site there has been eating. We need to screen that there should not be have the any off target. Once these animals will be born, we need to perform their health, how the growth rate is really improved or not, or their health, how are they behaving the similar health to the non-edited animal, that is a control animal or conventional breed animals. This is our the recent the program what we have been working. So I think why we have been working on this, I think I will not go into detail, but just would like to highlight the buffalo is not only the milk animal, it is a meat animal too. If we talk, if we talk how much the money we have in the Indian government or the people they are earning, those are involved in the meat industry, particularly for the buffalo meat, they have been every year more than the 20,000 crore rupees. So we also not only rear the buffalo for the milk production, but there is a need to rear these animals for the meat production too, because there is a huge demand, and these particularly meat from the buffalo they have been exported. The 99% of the meat from the buffalo has been exported to the other countries. In addition to that, I think everybody knows that we have the huge demand of the meat from the sheep and goats, so that's we also been targeting. And if we talked about the assisted reproduction as well, I'm talking, there's all this technique like the in vitro maturation of those site or IVF for the cloning, then well optimized in the buffalo. We know that buffalo produce many clones, or the IVFs also, and the sheep and goats also. Our partner from the CARG or Cost Kashmir, they are well established protocols of this technique. Now, what we have been currently, we have been developed the vectors already. We have been midway. We are not yet transferred these embryos, but we have been midway. We have developed the animals. We developed the CRISPR guides again, the myostatin gene, and then we also optimize the electroporation conditions. These are the different electroporation conditions we optimize for the buffalo somatic cell. This is study from the buffalo, and different concentration of plasmid, 2.5, 5, and 10 microgram, different size of cuvettes, as well as what we get the. But despite that, currently we have the maximum transfection rate is a 15%. This is much lesser. So what I believe is that because the vector size is also an important. In this case, our vector size is the more than the 10 kb, around the 10 kb. So that vector, so if you the larger vector size, then it is very difficult because the pores form through the electroporation or even from the lipofectamine. It's really difficult to transfect this large size vector into the buffalo cells. It might be the cell type also been one of the reason. But in addition to that, we also been used to generate some of the transgenic cell line using the transposon approaches. That is the transposons have been used, and we generated in our laboratory the 4 transgenic cell lines, and this is from the cell cloning, single cell cloning buffalo. We optimize, this is for the red transgenic cell line with the M cherry, which has an express in buffalo cells, and this one is a green transgenic cell line expressed in the venous reporter. These are the reporter cell line. In addition to that, we have been also worked on the myostatin knocking out. These are the just 10 pictures of the bright light pictures of the buffalo cells under the fluorescent microscope. They have in different transfections rate. If you look at here, this particularly, these are the CRISPR2, these are the CRISPR plasmids. So CRISPR plasmid, as I'm talking about, this very few the green cell is there because the vector size of this PF4545 is in freely it has been available on the gene. So if anybody wanted to order, they can approach the gene to have this plasmid in the laboratory. But if you look at here and here, these are the transposons plasmid. These are the size of the transfer plasmid around the 5 or 5.5 kb. So because the smaller size here, the transfection rate is higher. If you look at 60% more than, and here also the 60%. So similarly, we have generated the transgenic cell lines for the 4 transgenic cell lines for this one, and for the myostatin, we have around the 9 genome. We have been screened them. We generate the 9 lines. Now we have been targeting to screen them to how they have been proper editing as well as off target. So this is one of the experiments we have been says that the mutation detected. We detected the mutation that they are really edited or not. If this is a T7E assay, I think I will not go into the detail about the how T7E assay is that. But just if you look at here, there is a 2 bands. Here is actually the 3 bands. This one, this is the, if you look at here, this is a positive control, this is a negative means without enzyme, and this is the with enzyme. This is untreated cell, this is a control cell, and this is a treated cell, transfected cell. If you look at without enzyme, because without enzyme there is no enzyme in, there is no cutting. If there is editing happen, editing means there is a deletion of some nucleotide, then there is a cut. There is a cut means there is a three bands will form, and these three bands with the cutting of the enzyme, and that clearly shows. If there is a no editing, for example, untreated cell, even if you add the enzyme, there is a no cutting. For example, here in the last second last well from the left, it is in here. If you look at here, without with enzymes also, there is a no cut in the lower band size, which indicated that our cell, what the protocol we have been optimizing buffalo, that been giving the proper editing. And that these cells, now we have the 7 8 cells, we have been sequenced them also. I have not been included in the data. We sequenced them also and shows that they have the, some cells have the two nucleotide chain, some cells have the five nucleotide chain, and some of the, and one cell line have the around the 17 nucleotide deletion. There's a chain mean deletion is there. So we expect when we use these cells as the nuclear donor, then these embryos when transplant, we expect that the animal produced through the use of this donor cell will have the improved meat yield. Now the another project, this is Indo-US project uh, with me, and in this project, what we have been aiming to produce the animals or develop the animals which has resistance to the Anemia hemolytica. I think those are been working in the health aspect. They knows that Anemia hemolytica, it's called the posturelosis, or a similar category to the HS, the hemorrhagic septicemia, but there's a Posturella multocida. Earlier, this bacteria is called the Posturella hemolytica. Now they have been naming as the Anemia hemolytica. So what the Anemia hemolytica, this disease is cattle and buffaloes, both they have been susceptible to the disease when there is a down in the immunity. This is in common cells bacteria in the respiratory tract. When the immunity down, either in the transport stress or the environmental stress, any kind, because this is posturelosis, the kind of infection, respiratory infection happens when the animal in distress. So many hemolytica, they will have their pathogenesis, and the pathogenesis into the lung and the damage to the lung under the stress condition, and the animal, once the animal develop the disease, then animal succumbs to the death. So, and this is one of the important economical disease. I see many of these animals have been died because of the hemolytica infection. So what we have been target, we want to do, and the previous studies, particularly not buffalo, is not been done, but in case of the cattle, it has been shows that this paper is published in PNAS. So it shows that the Anemia hemolytica secrete the leukotoxin. Leukotoxin means a toxin that target the leukocyte, and because in ruminants, it's very interesting to note why the ruminants are, the non-ruminants are not infected to the this Anemia infection, for example, pigs or other. Okay, so why they have been identifying is that in ruminants, particular ruminants, the cattle, sheep, and goat, they found that in leukocytes of ruminant they have been changed. There's cumin glutamic acid replace glutamic acid is there in fifth position of the signal peptide of the CD18 gene. This is a CD18 gene. I think CD18, you know, people that they have been receptor on the there's a leukocyte or lymphocyte, but because of that, the glutamic acid, which is present on the minus fifth position, means upward position of the CD18 receptor, and because of that, this signal peptide, I think in any protein, they have the signal peptide to transport them. So signal peptides are intact on the leukocyte, and if there is intact on the leukocyte, the toxin, that is the leukotoxin, or it's a name as a toxin, we say is a toxin secreted by the Anemia hemolytica. It will bind to the leukocyte receptor, that is the CD18 receptor, and that cause the leucolysis. The leucolysis is caused, and ultimately that will cause the acute inflammation because it is an immunity cell, and they will down the immunity and damage to the lung parenchyma and animal succumbs to infection. What we have been thinking now, if it's possible to replace this glutamic acid with the glycine. In case of the non-ruminant human, the glutamic acid is there instead of the the glycine is there instead of the glutamic acid. This is a similar situation if you link to the sickle cell anemia, where the valine is there. in case of the hemoglobin residue, which has been sickle cell disease. So here our aim was to replace the glutamic acid with the glycine, and we wanted to use the CRISPR tool. So in this project, what we have been currently been, we have the vectors we are designing. We transfect them. First, where we generate the vectors, 6 vectors we have been generated in our laboratory. We transfect them, we optimize them, and now we have been optimized that there is editing is there. So that is the CD18 gene is the editing has been happened. Now, what we have been next been targeting, we will provide the correct nucleo, that is a sequence which have these glutamic acid, means the a nucleotide which is a code for the glycine in the donor template, so that when they have been doing the editing on the CD18 gene receptor, particularly for the signal peptide, the glutamic acid will replace to the glycine. This is one of the important project in addition to the what the myostatin we have been doing to one project for the improved meat yield, and this is this project I really wanted to have this the kind of the mutation so that the glutamic acid need to be replaced with the glycine. This is also we are working on this. One of my PhD student is taking this project, and she is in continuing on the in case of the cell lines, her work until the generation of the cell line. Now coming into the regulatory, this is all about the what we have been from the last 2 or 3 years. We have been initiating the buffalo. This is a genome editing work in farm animals like the sheep, goats, and obviously in buffalo. In addition to that, I will not go into the detail because when I think in my next talk, I will talk more about the regulation and the ethics about the all the kind of the gene manipulation. But just would like to highlight, I think those who wanted to work on the either in pathogenic bacteria, virus, they need to follow the guideline. Institutional biosafety committee is there in every institute. After that, there is a RCGM committee is there. So you need to take an prior approval if you wanted to work on these techniques. There's a gene editing techniques or gene techniques. So there is a group of the animals. I will go more detail in my next talk, but just would like to highlight is that our current these the projects over the group 1, group 2, and group 3. So doubling muscle mass, our the project has been fall into group 1. It will easy to get the approval from, and we have the approval for these. Previously, I have the approval for these double muscle mass animal production. Tuberculosis resistance, for example, the more deletion or more manipulation of the gene into the genome, this will fall into the group 2 category. And group 3 category, in which, for example, I was talking about the insertion of the human insulin gene into domestic animal, it's a fall into the group 3 category. If anybody wanted to have worked on the all the kind of the gene technology, they need to understand which category. Particularly, this is particularly for the gene editing. I'm not talking about the gene cloning or the viral infections or the bacterial diseases. They need to see, but despite that, you need to have approval from the IBSC and RCGM. I will talk more about my next talk. So now, what is the regulatory? This is in draft proposal proposed by the DBT. As by talking, there are the 3 committees which has been regulating the gene technology work. One is the IBSC Institute Biosafety Committee, RCGM. This is the statutory body at the or advisory body at the Department of Biotechnology, and the GEC. This is the approving body at the Ministry of the Environment and Forest. And without the approval, if we have to develop the technology, then you need to take an first we need to take approval from the IBSC, then need to be the RCGM, and final approving authority to have the research experimentation is by the GEAC. This is the Genetic Engineering Appraisal Committee at the Ministry of Environment and Forest. This is the stage for the particularly for the GE animal. And if you have the animal used and wanted to or finally wanted to use in the field or wanted to translate this technology into the field, then ultimate final authority is the Department of Animal Husbandry and Dairying, which is a department now the Ministry of the Government of India. So this is then regulatory approaches. Now, what is the current research project in? Just I will sum up what we have been working. So I will sum up the projects I have not been discussed. This is only important project. 2 projects I have discussed. One is the doubling muscle mass animal production in buffalo, sheep, and goats. Another is a bovine respiratory diseases in buffalo. We have been working on this. We have also worked on the insertion of the human lysozyme gene into the buffalo mammary gland. This aiming want to protect the animals from the more infection, like the human lysozyme. This is the lysozyme has been protection against infection, particularly for the mastitis. If we have human lysozyme secrete into the mammary gland, then assumes to expected that the animals are resistant to the mastitis. They will not have the mastitis. Another what we have been working on unraveling, identifying the role of the developmental genes. Suppose if you're knocking out the gene, particularly for the embryos, like the OX, SOX, and NR, or other genes which are responsible for the developmental genes, we also been knocking out some of the genes and to understand that if you're knocking out the, for example, OC4, is that embryos developed the blastocyst or not, and how these genes are regulated the embryogenesis. This is the basic work we have been working. There's a couple of areas we have been working in our laboratory. So in the sum of my talk is that the any trait can be treated by the CRISPR. It is either production related trait or health related trait. But what we need to identify what actually India needs, I think we need to understand that what kind of the research we need to pursue. Suppose we in India, if you are agricultural science, we talked about the majority of the people they wanted to have to develop the breeds or varieties or improve something. But for the basic research, for to understand something, there's a very less funding has been diverted to understand the basic mechanism or the pathogenesis or the gene function. But there is a requirement which we need to understand what actually India needs. Obviously, we have the highest opportunity because we are being going to be the most populous country. We have the huge young people. We have the now been government has been because of the COVID or because of the funding has been shrunken to these agricultural science. But hope is that we have more opportunities. We have diversity is there. We have more crops, more breeds of the animal. I think India is the only country which has the highest diversity in the. I'll be talking about the animal population. We have the huge number of the breeds and all the species like the cattle, sheep, goats, buffaloes. So we need to tackle these all the kind of opportunity. Despite that, we have the highest challenges too, because in working Indian condition is a bit difficult. We some of the compromising with the quality infrastructure we have. Sometimes we compromise with the quality manpower we have. So we need to think that what kind of the challenges that need to be understand, and with that we will succeed to the all any kind of the research in the world. This is the before the end of my talk. Uh, I would like to acknowledge the obviously the funding because the funds are really important. Uh, which has been important source to continue the any kind of research. Majority of fund is coming from the ICR, which is our sole funder. But in addition to that, Department of Biotechnology, Department of Science and Technology, SERB, or the Indo-US Science and Technology Forum, and DAG, and my previous institution, where I've been initiated all this works, and now I'm continuing at the NDRI, the Central Institute of Research in Buffalo, for their support to continue this kind of work in our laboratory. Thank you all. If you have any question, I would like to take it. Hello.
Sir, thank you very much for your enlightening talk. It was a very interesting talk on CRISPR especially. And now I request the audience to please send in their questions to Dr. Sonal Ingre. If she has got any questions, Sonal, did you receive any questions?
Madam, there are two questions in the chat box. Yeah, one is like Dr. Savita is asking, what's the status of gene editing in poultry?
If we talked about in India, there is a poultry research institute, and Dr. Bhattacharya is there. He is working on the poultry gene editing in India, but we have not yet produced in India any gene editing animals, either in poultry or crop. But if you talked about the abroad, the people has been already produced the gene edited poultries for the diseases, particularly leukemia disease is there. So avian leukosis, that is called the avian leukosis. So they have edited the gene responsible for causing of the avian leukosis, and these poultry are not infected with the leukosis. Also, for the meat yield, this myostatin gene is knocking out and then improve the meat productivity. Overall, globally wise, there are many advances been done. If you talk about the India, very few people they have been worked on the gene editing in the poultry. And as I know, only Dr. Bhattacharya, he has been working on the gene editing in poultry. Rest, maybe I was not aware of it in India.
There is one question from Dr. Charlotte. What is your opinion on cloning in humans?
Cloning in human, it's a ban. I think I will talk in my next talk about the legal and regulatory issues of all the assisted reproduction, cloning, gene or gene editing. In human is a completely banned. There is a not possible, particularly in human also. If you wanted to work on the human embryos, you are not allowing to culture the human embryos more than the 14 days in vitro. Like when IVF babies are there, so these embryos need to be transferred not more than 7 days. So after 5 days or 7 days, these embryos need to be transferred. You cannot culture them more than the 14 days. Also, in cloning in human is completely banned globally.
Okay, and then there's one more question from Dipankar Paul. Can we validate CRISPR or Cas9 editing in transfected cells with the help of Sangha sequencing results?
Yes, yes, yes. So for validation, Sangha sequencing means you need to first you need to amplify the target site, and this target site, for example, with the PCR, you need to amplify this 500 or 600 nucleotide. Then this fragment of the PCR need to be the clone into the cloning vector, then transform them into the bacteria, pick up the colony, and this 500 600 target nucleotide will sequence to the Sangha sequencing. This is one of the approach. It is quite possible. If you wanted to completely characterize or screen, then you need to go for the whole genome sequencing.
And one question from Dr. Gaurav Datta. Any recent developments or projects undergoing in cattle in India through CRISPR, and what is the cost efficiency?
Currently in India, in cattle, we are means as far as know, it's nobody is working on the cattle, because in India the problem with the in cattle working with is all the transgenic or assisted reproduction or genome editing. We have the limited we have no availability of the slaughterhouse material because cow slaughter has been banned everywhere. So to have the all the kind of work, we need to have the oocytes or the biological material oocytes, particularly for the oocyte to produce an embryos. So in currently we is not available, but because now there is OPU and IVF technique has been done, we have been initiated in the NDRI to initiate OPU, IVF, and the cloning cattle cloning in India through the oocyte received from the home pickup. So I hope, I think in future there is a possibility to have the genome editing or even the transgenic work in the cattle. But currently there is no one working on this aspect.
Any more questions from the participants?
I think one participant has sent the question in the chat. I just saw it. I don't know whether it's really a question or not.
What is the status of double muscling in goat?
Yeah.
In double muscling in goat is already produced in many countries except in India. We have not yet produced, but our current project is aiming to have the double muscling in goat. But in all other countries, there is already produced. And the progenies of the double muscle animals, they have also have the 20 or 30% of the improved growth rate, the double muscle character.
Right, sir.
Dr. Ingole, can I intervene in between?
Yes, yes, sir.
Very good morning to all. Good morning, sir. I can see one more question here. If you want to conduct a genome editing experiment in vitro animal cell culture, then which authority's approval is required?
For in vitro culture, IBSC Institute Biosafety Committee is an authority to approve your experiment for the cell culture. I will talk more detail about in my next presentation.
Right.
IBSC Institute Biosafety Committee.
And
For in
could we
vitro
become...
work, IBSC approval.
Okay, sure. So, dear participants, stay tuned. So, your answer will be, or your query will be answered in the next session. We can have the last question here. Please discuss the cost efficiency of CRISPR. We know it's cheaper, but in little details.
regarding the cost efficiency, still I think nobody has been calculate the cost efficiency, because this technique not kind of technique everybody is going to use. It technique, this all this technique is high-end technique, will be used either the laboratories which have the facilities or the commercial companies. There was aim was to produce these genetic stocks of this animal. So this will not ultimately reach to the means there the product will reach, but the technology will not reach to the ground level. For example, we thinking about the farmers, it will not reach to the farmers. Even the cloning or OPU and IVF will not be ultimately used by the farmers. It will be used by the big commercial companies or not. But still, if you're talking about the cost of gene editing, it's been high as compared to the other technique like the OPU and IVF, which is lesser been cheaper. For example, AI is much cheaper. Is compared to the AI, OPU and IVF is costlier. Is compared to the OPU and IVF, cloning is much costlier, and compared to the cloning, the genome editing is much costlier. This is the way I think I would like to uh, share my view.
I presume there are no more questions in the chat box now. Madam, can we move to the next, please? If you kind
I
permission.
think Dr. Vaskar, if Dr. Selokar has finished, we go on to the next speaker.
Yeah, definitely. We can.
First, summarize Dr. Selokar's work, I think, right at the end, because there is many things which will be connected in his next talk also. So we'll summarize his talk later.
Yeah, sure. Thank you. Thank you, madam.
Thank you.
Thank you.
Thank you, sir.
Thank you, Dr. Selokar, for your informative presentation. Before moving on to the next speaker, I request all the participants to take care of your microphones. Please mute your microphones. It creates a lot of disturbance for the speakers as well. Now, moving on to the next speaker, we have with us Dr. Jay Kaushik, Principal Scientist, Coordinator, Biotechnology Information System Subcenter at the Animal Biotechnology Center, again from ICAR, a National Dairy Research Institute at Karnal. Sir has completed his BSc from the University of Delhi, and then he pursued Masters and PhD in Biotechnology from JNU. He has completed his PDF from the Institute of Protein Research at Osaka University, Japan. Sir is specialized in gene cloning and expression, biophysical chemistry, structural biology, and molecular biology. He has also been trained in computational biology in the laboratory of a Nobel laureate, Professor Arya Warshel. Current research is basically focused, his team focuses on the animal originated antimicrobial proteins, recombinant proteolytic enzymes, again from the organisms and the animals, then proteins associated with animal reproduction and health, and animal proteomics. Sir has 8 awards to his credit and numerous publications in journals of national and international repute. So without taking much time, I invite Dr. Kaushik sir, and again hand over the session to Dr. Maji ma'am and Dr. Vaskar sir. Please.
Yeah, thank you for my introduction.
Now call upon Dr. Kaushik to kindly start his talk.
Thank you very
Thank
much.
you, Dr. Kaushik.
Hope my slides are shared now.
Yes, please. Yes, please go ahead.
Okay. So in today's talk, I'll be talking about the recombinant gene expression in E. coli and yeast. Basically, this work, what we do in our laboratory for biophysical and structural characterization, as well as to understand the functional properties of the proteins. And as Dr. Naresh Silokar has already delivered his talk on CRISPR-Cas, that's much more advanced, and that's in continuation of what I will be talking about. So his system is just directly manipulating the genome. So what I will be talking about, how to manipulate the E. coli or any bacteria for that matter, or you can say yeast or plant cell or insect cell, or you can say mammalian cells. So you can manipulate all these to express a gene of your interest and produce the recombinant proteins. And that can be used for diverse functions that I'll be talking about. Like why to produce recombinant proteins. Recombinant proteins, because they have immense applications, like what has been there in the past. Study structure function relationship in protein. Like we didn't know how the protein works and which particular residues are important for the activity of the protein. So by using the recombinant DNA technology and combined with the mutagenesis, side-directed mutagenesis, you can carry out a mutations and check which particular amino acid is responsible for the biological activity of the protein. So that type of study we can carry out, although this used to be done by chemical modification of side chains, but that is not perfect, because if you try to modify a lysine, for example, then all the lysine will be modified. So you cannot make it out the effect of change of lysines or modified lysine is coming from a particular lysine or for all the lysines. So likewise, cysteine can also be changed, but by side-directed mutagenesis, you can directly pinpoint and mutate a given residue and change the sequence of the protein and see its effect on the structure as well as function and stability and conformation. So all this type of work we carry out, and then study biochemical and enzyme properties of the protein, then exploit at industrial scale for biotransformation. For example, in the bagasse or paper and pulp industry, huge amount of endogluconases, gyleanases, cellulases are required. So for that type of work, we can produce large amount of recombinant proteins, which is not available from the native protein, native source. Then produce bioactive peptide and biopharmaceutical. So like insulin is the best example that has been used from the recombinant sources. Likewise, we have these vaccines for the COVID that is also recombinant. Produce diagnostic kit for the detection of antigens, antibody, active ingredients, pathogens, xenobiotics, or any kind of analyte. What it means that for diagnostics, it's much easier to use this technology rather than producing the vaccines. So this is the first big population study we are doing on COVID, that we are using the recombinant proteins at such a large scale, and they have been very successful. But for diagnostic purposes, a lot of work has been done and many proteins are being used, or even for production of biosensors as well. So we use the recombinant proteins to design these switches. Not only this thing, like many of you must be knowing the green fluorescent protein, and this green fluorescent protein can be converted into a red fluorescent protein, yellow fluorescent protein, just by few amino acid changes. So this is by genetic engineering or protein engineering, what we call it. Then construct biosensor and electronic devices, which are people are trying to produce very fast switches by using the switching on and off, like phosphorylation and dephosphorylation. So phosphorylation means switch on, dephosphorylation means switch off. So this type of thing can be carried out with the proteins. Then produce cleaning agents, like in the industry, like for example in dairy industry, we have lot of fat and proteins which get accumulated around, or like in the cleaning of the utensils at the home, we have to produce some enzyme, and that enzymes can scrub. They are very effective. And also in many chemistry laboratory or industrial scales processes, they are being carried out by using the chemical methods, and those chemical methods require huge amount of chemicals, like organic solvents. And those organic solvents, for example, if you start with 100 of liters, then your output is something a few kilogram or few liters, or even less than that one. So the efficiency of chemical reaction is very low, but because enzymes, which are proteins, they are highly specific, you can reach to almost maybe 100% efficiency if your Km value, that is the Michaelis-Menten constant, is very small. So one can try to improve the Michaelis-Menten constant of the enzymes, can improve the kcat of the enzymes, that is catalytic rate, or you can try to stabilize the proteins. For example, we have the many enzymes which get denatured if you bring it at the room temperature. And therefore, to stabilize them, we had to carry out the protein engineering that we go through the genetic engineering. And likewise, vaccines. Why vaccines fails? Because they are not maintained in their native state against which the antibody are raised. So therefore, if they denature, then their conformation change. And if we have the antibodies are conformational dependent, then what will happen? Then this antibody will bind to the right antigen because the antigen has got unfolded. And that's a big problem in countries like India, where we do not have the refrigeration system or to carry out these vaccination plan. Therefore, polio vaccine also failed because of non-maintenance of the temperature. Likewise, many biopharmaceuticals require the refrigerations or low temperature, and therefore, protein engineering or genetic engineering has a big role in stabilization of the enzymes. Think of, we use restriction enzyme or ligase in our laboratory in manipulation of the DNA. If we keep it at -20 and some student comes and use it, and let's say he forgot it outside itself and it remained outside in the laboratory all of the night, and next day somebody will work on this one. So his or her experiment is liable to be failed because protein is already denatured partially or completely. So therefore, they may wonder why their system is not working. So just to work out this thing, it can consume weeks or maybe months to figure out what is wrong, because we will think that our protein is problematic. We'll think that something is wrong with our the way we are doing. Maybe our buffers are problematic. We'll set the pH. We'll check the ionic concentration, and all these things will carry out, because the first thing we think is something wrong with our carrying out the experiment. So, but at the end, we will find that problem is the enzymes. So if enzymes are robust, there are many advantages of this thing. For example, in biotransformation of the reactions, you can see that a fermenter or a biotransfer vessels is being carried out, let's say, at 25 degrees Celsius. If you raise the temperature to higher, let's say 30 or 35, then the enzyme activity may go down, or its life will go down because of kinetic problems. Now think of that you can increase the stability of this enzyme to 40 degree, where it can work very well. And you are running a fermenter at 25 degree. So what will happen? That temperature may fluctuate. If it is highly susceptible to the 25 degree, like if you go to 30 or 35, what will happen? It may partially lose activity. So, but if it is stable, it means you need a chilling plant. So you need these circulators, water baths to cool it down. That needs a lot of energy and additional investment. But if you have an enzyme which can denature, which will not denature at 40 degree or 50 degree, then whether your temperature is 30 degree, 35 degree, 40 degree, it is not going to denature. If it is not going to denature at 40 degrees Celsius, then think of from the basic knowledge of chemistry that if you increase the temperature by 10 degree, then the rate of reaction will be double. So same may be true with the enzymes, but not exactly follow the same rule. But of course, when you increase the temperature, the activity of the enzyme will be increased. And another advantage is that some substrate, they may not be soluble at 25 degree or 30 degrees Celsius. If you raise the temperature to 40 degrees Celsius, 45 degrees Celsius, it may be very well soluble. And one big advantage is that contamination. So if you are carrying out a reaction at 25, 30, 35, then it's easier to get the contamination in the vessel. And you may require some antibiotics, and that requires some additional cost. Let's say your enzyme is stable up to 60 degrees Celsius. Now you can carry out a reaction at 45. If you're carrying out a reaction at 45, chances of contamination are reduced tremendously as compared to when you do at low temperature. So there are several advantages of using a thermostable enzymes. And all these things can be carried out or produced using the recombinant DNA technology or by through protein engineering or by recombinatory mutagenesis. Combining all these things, you can produce very robust enzymes. So what are the steps for this thing? So the initial steps usually I teach to my student as well. So it is more of like a teaching type of rather than like a presentation. So considering all these factors, we will be going with the very basics, because I understand not all the people are from biotechnology. They are not studied probably what is the recombinant DNA technology, which I have been told. If it is like this thing, I will start with the very fundamental things like what are the heterologous system for gene expression. We have bacterial, easiest most. Then you in that one, the most easiest is Escherichia coli, E. coli, because this is, we know the genetics, we know the biochemistry, we know the molecular biology of this organism, so we can easily manipulate. If you can easily manipulate, you understand if something goes wrong, you can understand where things have gone wrong, and you can try to improve that one. And there are many hosts, because there are specific requirement of the host to produce the recombinant proteins. So all these manipulations are to be carried out so that genetically in the E. coli host, so that it can produce large amount of the protein without digesting it, because a protein which is being produced can also as a source of nutrients for the same organism. From the host. Then we have the Lactobacilli, that is a dairy organism. People are trying to find how to engineer this one or how to make it to produce the proteins. And Lactococcus is very well-known organism, Lactococcus lactis, which has been used for genetic engineering of purposes. Then yeast and fungi. So they are very robust one. And added advantage is that in comparison to bacterial is that yeast and fungi, they are eukaryote, although lower eukaryotes. They cannot carry out all the function which are being carried out by the higher eukaryotes for the post-translation modification. For example, if you are trying to express a protein which is coming from the higher eukaryotes, let's say from the human, animal, like you say cattle, buffalo, or anything. So if you are trying to express the protein in the bacteria, those protein or that gene product has never seen an environment of bacteria. So it may not fold properly into its three-dimensional structure. If it cannot fold in its three-dimensional structure, it cannot carry out its desired function. It means the whole of the idea of producing the protein will fail in bacteria if it cannot fold properly to a biologically active protein. So therefore, if we go to the yeast, if that fails, then yeast and fungi, they are better adapted to produce the eukaryotic protein, because there are also eukaryote, although low eukaryotes. In that case, we have Saccharomyces cerevisiae, which is very well known, Cerevisia pombi, then Picia pastoris, Picia methylotropica, and Sinula polymorpha, and Chloromyces lactis. So Chloromyces lactis is a dairy organism, which is very similar to Saccharomyces cerevisiae, but adapted to dairy system. But we in our laboratory and many other laboratory, they use Picia pastoris here, this one. Then in the fungi, we have Aspergillus niger, Aspergillus nucor, Aspergillus avamori, then Candida boldini. So people have already developed the system. When I'm talking about the expression system, what I'm talking about the host and plasmid. The plasmid which will carry on your insert, where you will clone your gene. And once the gene is ligated with your backbone of the vector, it will be transformed into the bacteria, yeast, insect cell, plant cell, or mammalian cell, wherever it is required to be expressed. It has to be transformed so that it will go there, will survive as an episomal molecule like a plasmid, as an independently replicating or autonomously replicating extracellular, not extracellular, extra chromosomal DNA. Or it may be, you may need it to be getting inserted or integrated into the genome so that it is not lost during the subsequent division of the cell. Then we have the insect cell. Many mammalian cells have been produced in insect cell as well for veterinary purposes, and all the people have produced even edible vaccines in plant. Sorry, I'm talking about the plant. So the plant are very good. Their glycosylation is not exactly similar to mammalian, but of course they are also high eukaryotes, so can be used for production of some protein whose function does not depend on post-translational modification. Then you have the insect cell. Insect cell are very close to mammalian cell, but not exactly mammalian cell. So they can mimic the protein produced in the insect cell can mimic the protein produced in the mammalian cells or like the native proteins. And you can produce large amount of proteins in the insect cell. So we have the baculovirus system, very powerful, can produce large amount of the protein. But of course, like the mammalian cells, we have the problem in the culturing of the insect cell. Then if none of the product produced in plant, insect, yeast or bacteria, they are not suitable for end use, then you have to go finally to the mammalian cells. A particular cell line may be required for the production of that one. But usually we can work in any mammalian cell because we express our protein under the powerful viral promoters. So if glycosylation or any post-translational modification is important, then in that case, we have to go to the mammalian cell. I think somebody is speaking in the background. May I request to please switch off your mic? How to choose an expression system? That is always a question to any student or any researcher when they are going to express a protein. Because I mentioned all these things. Bacterial is the easiest because its doubling time is 20 minutes. Yeast, it 66 hours. Okay, approximately 6 hours. So the accumulation of biomass will take longer in yeast as compared to bacteria. The bacterial system is so fast that once you start in induction of the gene expression, within 3 hours you can collect sufficient amount of the protein. But in yeast, it takes at least 48 hours or maybe 2 or 3 or 4 days for production of the protein. And insect cell and mammalian cells, you know, it's take pretty long and they are very susceptible to infections, and there are problems in those ones, but still may be required. So how to choose? How to find? Should I straight away go to the mammalian system, or I should start first in the E. coli, then fail in E. coli, then go to the yeast system, fail in yeast system, then go to the any other system like the plant system or say something like insect system, fail in that one, and then reach to the mammalian system? Or we have some idea. If you know your protein is likely to function even without glycosylation, or there is no glycosylation or post-translational modification. What does it mean, post-translational modification? Glycosylation is a post-translational modification, as well as proteolysis. For example, insulin. Insulin is produced as one big molecule, but it is digested into alpha and beta subunit by a proteolytic. Likewise, many proteins are produced as a inactive molecule, which are converted to an active molecule by proteolysis. That is called the post-translational modification. If that is not possible in your system of interest where you are trying to express your protein, then probably you have to move ahead with another system which can carry out this function, glycosylation, even phosphorylation, or some particular specific type of glycosylation are possible in only mammalian system. A yeast can carry out simple, like it can carry out tag mannose, but not very complex type of glycosylation like sialic acid tagging, and all these things are not possible. Now, if the native protein is a protein, glycoprotein, then post-translationally modified protein or which has a folding issues in E. coli or lower system of your expression, then the system of choice should be which is very close to the system source of gene. If you are, it is a mammalian, then probably it is better to go for the mammalian first. If the product is a biopharmaceutical, for example, an injectable drug or a vaccine, for end use in human, animals, or then the expression host should be relevant host cell line or mammalian cell line. If the product is for diagnostic purposes, because you are not going to use it for human use, you will analyze human or animal product in vitro, not in vivo. So therefore, you can produce your protein in any system in which you are comfortable. Then if the product is likely for industrial purpose, the host could be E. coli or yeast fungi, depending upon the activity of the protein. And then if product means protein is needed for academic studies, one can need to see how PTM affect the biological activity, or if one want to understand the structure function relationship, then choose a system for easiest manipulation and alteration, because you have to carry out many mutations again and again in different residue to understand which residue is responsible for which function. Or if there is a PhD student, he or she has to carry out some manipulation in the protein sequence, or just native protein sequence has to be produced, then straight away one can go to the E. coli level. But it should be active. But if it is not active, then you have to go to even to mammalian system. Then engineered yeast or fungi would have attenuated PTM to mimic high recurrence. Now these days, people have engineered yeast and fungi which can mimic glycosylation similar to the mammalian cells. So, but they are very expensive, but they are still available, and people are making more complex system for the production of high eukaryotes protein into lower eukaryotes or in prokaryotes. Now, the first and foremost requirement for the expression of a protein is cloning vectors or expression vector. So the difference between cloning vector and expression, major difference is that expression vectors should have some promoter. Okay, so the promoter which will drive the expression of your gene to produce the transcript, those transcript will be used for production of your protein through the translational machinery, that is the ribosomes. While in this case, there is no need for promoter. Of course, usually different companies, they always place some promoter, but those promoter are not meant for driving the expression. So these cloning vectors, they usually are high copy number, while expression vectors are low copy number. Low copy number because if you use copy number, for example, 200 copies or 300 copies number of expression vectors, they can be produced in E. coli, then there will be equal number of your insert which will be expressed. Along with your insert, there are few other proteins like marker genes, like antibiotic resistance gene marker will also be expressed from this backbone. It means so many number of copy of the same will be there. It means RNA polymerase of the E. coli or your host will be diverted toward the production of transcript of only this one. It means it will be bereft. It will not have sufficient amount of RNA polymerase for its maintenance, for its propagation. It means it may die out. It may be killed because it cannot compete. It is not healthy enough to maintain itself. So therefore, expression vector copy number is usually maintained at low, that is something like 20, 25 or even less than that one, because we don't want to maintain hundreds of copies of your gene which will be transcribed. And there are some other problems of over transcription, that your protein will be produced in huge amount and it will carry out the crowding in the host, and that crowding will have its own effects. And but here the cloning vector is mainly to clone, to amplify, or to maintain your gene construct. So therefore, in few cycles of division of your bacteria, you would like to have huge amount of plasmid. So even if with few cycles you can produce large amount of the plasmid, then that is better, because you are not going to express your protein. It means it will take the enzymes which are responsible for the replication, but not which are responsible for the transcription and translation. So usually we use this PZ 1.2, that is a blunt end. So this is called TA cloning vector. Here one T and another T are present because when you produce a product by your PCR, that place one A and A on both sides of the fragment, and that AA will ligate with the TT here present, T here and T here on that one, and by that way it can ligate. So this is called TA clone, while this is blunt end clone, and you have many numbers of restriction enzyme. You have to clone somewhere here. This is the multiple cloning sites. Here is the multiple cloning sites. So you will clone it here. So if you clone it here, you clone anywhere with any restriction enzymes, but you can release the product by using any of these restriction enzymes. So another important thing in all these things is the origin of replication. The origin of replication is species specific. For example, if you are carrying out the translation, you want to amplify it in E. coli, then you should have the origin of replication, which is based on E. coli. Okay, here the ORI means origin of replication. This is the origin of replication. Here PBR 322 origin based sequences which are present in this particular vector. So origin of replication restrict the number. It decides how many copies number of the plasmid will be maintained. So the relaxed type and some are restricted one. Restricted means low copy number, and high copy where replication is allowed. If you go to vectors, so there are other elements in this one. You have P-tag. Here you can see there is a P-tag. This is promoter, which is called tag promoter, you can say. Then there is a glutathione esterase. This is a fusion protein. So your promoter will drive the expression of glutathione S transferase, and then you will clone your gene of interest here. It means you are going to produce a fusion protein on whose N terminal you have the glutathione S transferase, and C terminal you have product of your gene. Here it can be anything. So once they are expressed, you can remove that one, you can purify that, and after purification you have to digest with a protease. So a proteolytic site is also provided here so that you can remove glutathione S transferase from your protein of interest and purify your protein. Or you can purify first and then you can digest. So likewise, you should have a ampicillin resistance or kanamycin resistance or hygromycin in eukaryotic cells. So these are minimal requirement because this is required for the selection of your, that transformation has taken place and your gene or your plasmid has gone inside, or your transformation or transfection has been successful. Then there is a LAC-I gene. So this is a repressor of this P-tag, because when you are not interested to express your gene of interest, then this P-tag or this promoter should be silent. So it should not express your protein when you are not interested. Here. So when you want to keep it silent, then there should be an operator sequence here. With that operator sequence, there will be a repressor protein, which will repress the transcription. So that repressor protein will produce from this G. And as I mentioned, that between the GST and your gene, there is always a proteolytic site. Here is a thrombin cleavage site. It means you have to use the thrombin enzyme. Sometimes it is factor 10, or sometimes some enterokinase, or some other protein which can be used. So it means you should have some specific sequences here which can be digested by your enzyme to release your product. And then you have, so now your vector for expression. As I mentioned in the previous slide, cloning vector and expression vector. So here I'm talking about measure. You have the promoter, then you have this Sine-Delgarno sequence, this one. Sine-Delgarno sequence I'm talking about in E. coli or prokaryotes is a sequence where ribosome will bind. Students who know about the molecular biology, how translation takes place, they know what is the Sine-Delgarno sequence. Then there is an N-terminal tag. Sometimes we need a tag. Tag is a small peptide which will be tagged to produce your recombinant protein. They help in purification. Then fusion protein, it helps in, I talked about the GST. GST will be used for purification of your product by affinity chromatography. In a single step, you can achieve 90% purification, or N-terminal tag, something like 6x his tag. So histidine is repeated 6 times. So this histidine will bind to some chelating agent, and in single step using affinity chromatography, you can purify your product, which is cloned in the multiple cloning site here. And then there is a protease site, the yellow color here. So once you purify, you can release your protein from this one. So this fusion protein and N-terminal tag are used for successful transcription as well as translational process to start to give the initiation part, because sometimes we have the problem in translation of the gene you are cloning. But these sequences are well optimized. Another advantage of this thing is that these proteins are highly soluble in the system of in your host. But if your product of your gene, that is the protein you are trying to express, does not fold properly in your host into 3-dimensional structure, then this fusion protein can help it to fold, because it isolates it from rest of the, because this know how to fold properly, and this can help your protein of interest to fold into 3-dimensional structure. And it can be done by isolating individual molecule from rest of the molecule, because large number of molecules of your protein will be produced which are nascent polypeptide chain, not properly folded. If they are not properly folded, their hydrophobic groups will be exposed. If hydrophobic groups are exposed, then they can interact with each other. If they interact with each other, then they form dimer, trimer, and they form a big assembly, and they form an inclusion body. We call it the inclusion body. And those inclusion bodies are misfolded protein, which are not biologically active. So therefore, your whole of the experiment in production of a recombinant protein will go in the vain. So therefore, we always sometimes use the fusion protein. Fusion protein can help your protein to not make interaction with other molecules, but can intramolecularly fold into the right conformation. Like C-terminal tag, you can have C-terminal, N-terminal tag, you can have the C-terminal tag. Because sometimes what happens, a tag or a fusion protein at the N-terminal of your protein may cause loss of activity. So therefore, you may like to have the tag at the C-terminal and then the transcriptional termination. And before the transcriptional termination, there will be stop codon here. That stop codon is for to stop the translation, while this termination for transcription is to terminate the mRNA synthesis beyond that point. Then we have the shuttle vector. In the shuttle vector, something like E. coli. So this is a vector which can be used in E. coli and Pichia pastoris. So this PICH is coming from Pichia and OLI is coming from E. coli. So this is a combination of both. Why? Because we have this T7 promoter. This T7 promoter is coming from prokaryotic system or E. coli. While AOX, this AOX promoter is coming from the yeast. So there are two promoters in tandem, AOX and T7. It means if you transform this construct where you have cloned your gene here in the multiple cloning site, after cloning your gene in this at this site, you will have a recombinant plasmid, which is a shuttle vector you transform into a host, and host could be here E. coli. If it is E. coli, then RNA polymerase of E. coli will bind to T7 promoter and start the transcription of this gene which is cloned here. If it is not producing the right kind of protein in E. coli, then you can switch over, you can simply transform this construct into the yeast, that is Pichia pastoris, because in Pichia pastoris RNA polymerase will bind to AOX promoter, because this is yeast specific promoter. If this is yeast specific promoter, then it can be transcribed and translated. So therefore, the same construct can be used for expression in E. coli and yeast. So you don't have to construct it twice. And PARS, this is Pichia specific or Pichia autonomous replication sequence. It means this particular construct can replicate in Pichia or yeast, while col E1 origin, this origin is for replication in E. coli. So whether it is a vector for expression in E. coli, in yeast, in plant, in mammalian or insect cell, whatever it is, we always provide a origin of replication for in E. coli, because most of the manipulation of the DNA, like insertion or ligation, all these things are done in E. coli. Once ligation is successful, or you have inserted your sequence in this downstream of the promoter, then you transform E. coli, amplify sufficient amount of the plasmid, and when you have sufficient amount of protein, this plasmid then carry out your transformation or transfection of your host, which could be mammalian cell, insect cell, plant cell, or E. coli, whatever it is. So here are the minimal requirement of for expression of genes. Here are other factors for specific purposes. I think I'm taking a little bit more time, so I will go ahead with this one. Second thing is RNA polymerase in the plasmid. This is the most important because it is the RNA polymerase which bind with the promoter. So a specific promoter which will be recognized by this particular promoter, this RNA polymerase. So it is the combination of RNA polymerase and RNA polymerase and promoter sequence, which will decide what will be the host and also origin of replication, these three things. So this is a simple one, RNA polymerase, like alpha, beta, beta prime, and sigma. But in eukaryotes, they are much more complex one. So I will go a little bit faster. So promoters are conjured sequences, like you take different genes, all these different genes in E. coli or in any given species, they have conjured region, -35 and -10, because this RNA polymerase will sit and recognize some particular sequences on this promoter sequence. One is -35 and another is -10 in prokaryotes. In eukaryotes, it's different. Okay, so they are consensus sequence. It means it should always be T here. Here it should be T, like 69% of the times, or in 69% of the genes, you will have the T here, T here, G here, A here, C here, and A here by this amount of percentage. And at -10, so here 77% times it will be this one. If you try to change this one, then the activity of the promoter will go down. So that's why we call it conjured or consensus sequence, which are present in most of the genes. Why they are present? Because evolution has taken place like that one, that RNA polymerase will bind to these sequences very strongly and start the transcription. If binding of the RNA polymerase is not strong, then it can bind and dislodge, and transcriptional start may not take place. If it start, then it may be decomplexed. So therefore, these are important, the binding of the RNA polymerase. Then initiation. Initiation is the most important event. It may bind, but may not start the initiation. So for initiation, you should have these sequences, A or T, G or C. Out of this thing, A is C and A are preferred as compared to G. So the rate of transcription will depend upon binding of RNA polymerase here and initiation site, how good they are. Then there are promoters which are native type in these genes, but people found that sometime in the native systems we don't have the right kind of promoter which can produce large amount of the protein, because the native promoter, they are made to for maintenance of the organism. They are not there to fulfill our greed of the human being, because we want to produce huge amount of the protein for our purpose, and E. coli is not geared for that one. It means we can combine a part of two promoters, a part, for example, in trip promoter, tryptophan trip, and then we have the lect promoter, lactose operon, then trip operon. From these things we had taken a part. Minus 35 was close to the consensus, close to the conjured one, which is close to the ideal promoter in the tryptophan promoter. Well, in the leg promoter, we have the minus 10 region, but in leg minus 35 was not close to the consensus. Likewise, in tryptophan promoter, the minus 10 was not close to the consensus. So therefore, we took this part, which is close to the consensus, from the leg promoter, and this part from the trip promoter, and combine them to produce a hybrid. And now the hybrid is called tag. And see it, the wild type of promoter was showing this activity as a function of time. This is the production of the product, human growth hormone. But when you clone this human growth hormone under the tag promoter, which is the hybrid promoter, the production of your protein is so high, many times. Okay. So that is the advantage of using powerful promoter. It means this promoter is powerful than this promoter. It means it's become a synthetic promoter. Now, a cloning strategy for expression of lactoferrin, because lactoferrin is very important, and we are trying to clone the lactoferrin from the goat and buffalo and cattle, and trying to compare them, which one is better with respect to their biophysical and biochemical activities. So we isolated the RNA. From the RNA, we amplified our gene, which is 2.2 kb, then ligated it with the cloning vector, from the cloning vector into subclone into the expression vector. Then the expression vector was transformed and got the colonies. From these colonies, we got the confirm that our product is there in this one. And from this thing, we cloned our product, because lactoferrin is constituted of two lobes, N lobe and C lobe. So they are very close to each other sequence wise, and both are 40 kilodalton. Total protein is 80 kilodalton. So we wanted to know how N lobe affects C lobe and C lobe affect N lobe, because lactoferrin is an iron binding protein. If these N and C lobe are separated from each other, then what will be the iron uptake by N and C lobe individually, as compared to when it is a complete hollow enzyme where N and C lobe are together? So we wanted to see this one. So one is that you purify the lactoferrin from the milk. If you purify the lactoferrin from the milk, then you will have to digest. If you digest them, then digestion is not accurate, and you can produce N and C lobe of different sizes, as well as the yield of the digestion is very small. You start with 100 milligram of the lactoferrin and you end up with few milligram. It means accuracy is not there, non-specific proteolytic digestion could be there, as well as then that will affect the activity. So therefore, by genetic engineering, we want to produce exact size of N and C lobe. So therefore, we separated that N and C lobe. So here 1.1 kilobase pair, 1.1 kilobase pair, total was 2.2 here. Okay, so we clone all these things, total N lobe and C lobe, and express them here in E. coli. and then confirmed by ELISA, also this Western blot. So this is complete, bigger size, and this is smaller size NNC lobes. And then we purified it by 6S histidine affinity purification. And then later work was to characterize after production of recombinant protein, you have to show that it is biologically active. If it is not biologically active, then there is no use of overproduction or production of huge amount of protein. That is of no use. Then we showed that it can bind RNA. So as a function of pH, like at pH 8, 100% of our molecule were saturated. We start decreasing the pH. Okay, so when pH is decreasing in this fashion, then the iron is getting released from the N and C lobe and hollow protein. And here you can see this particular curve shown iron released by intact native LF. Curve with solid circle. It means this one is intact lactoferrin, while recombinant full length lactoferrin, open circle. It means we also purified from the milk and we produce recombinant. So their behavior is exactly similar. One is open circle, another is this solid circle. So their behavior is exactly same. It means we can say probably our recombinant lactoferrin is very close to the native protein which is produced in the milk of mammals. And then this is the iron binding property of the N and C lobe. Now C lobe open triangle, this one, and N lobe is this one. It means N lobe binds much more strongly than the, C lobe binds much more strongly than the N lobe, because in N lobe at pH 6, we have almost 50% release of the iron at pH 6. But in case of C lobe, we need pH 4. We have to decrease pH 4 from pH 6. It means at 50% of the iron or 50% of the population of the N lobe is saturated or desaturated at pH 6, while for C lobe it is only something like 35% is desaturated. It means 65% is still saturated. And to reach to 50% desaturation with the iron, we have to decrease the pH below 5. It means C lobe can bind more strongly than the N lobe. And this is the native and recombinant hollow. It means they are attenuating with each other. Okay. Then we also see the effect of this lactoferrin on the expression of beta-casein, kappa-casein, and interleukin-1 beta on buffalo mammary epithelial cell. How they affect, because lactoferrin is produced in the mammary gland. Whether they can affect the mammary epithelial cells in some way, because we have seen that at the involution stage, when the animal is getting dried, milking is getting dried, then the production of milk ceases. Then what happens? There is a remodeling of the mammary gland. We were trying to find out whether this lactoferrin can affect the remodeling, and we see that this is the control one in different media, hormone and hormonal media, while when we add lactoferrin in the culture, then the expression level is very, very low, right? It means it is affecting the mammary cell. It means it can help in remodeling of the mammary gland for getting it prepared for the next lactation. And here in this case, this is the cell proliferation. So here is the control, and when we add certain amount of lactoferrin, 0.1, 0.5 or 1 milligram per ml, then it affects the proliferation. It means it checks the proliferation. And some people are thinking to use it as anti-cancer agent, and people have used it for this one. Although the amount required is very high, but of course it has anti-proliferative activity. And our recombinant showed this one. But this recombinant was produced in P. pasteuris. So this was another protein that was a protein which was coming from the mammals, animals, and we were trying to produce in E. coli as well as yeast. And what we produced in yeast was functionally viable. It was having the similar properties with the iron binding and release, which is similar to the natively produced protein in the milk, as well as it was having anti-proliferative activity and other different activities we studied. So we are in the job, our student produce large number of proteins, each one having different kind of function. So we are also working on the probiotics. So our idea is that rather than using the probiotics are which we consume, go in our gut, and they protect us from the pathogens, or they can have some other immunomodulation. So therefore, we were having the hypothesis, rather than using the probiotic organism like lactic acid bacteria, or maybe some other, even E. coli sometimes function, some particular strain of E. coli also function as probiotic. Then some yeast also function as probiotics. They change our system. They immunopotentiate our gut system. They also protect the reproductive system, the vaginal system in the females. So we were of the hypothesis that rather than using the whole cell of lactic acid bacteria, which is a probiotic, because it is difficult to maintain. If you keep it at room temperature for a very long time, it may get unviable. You may not culture in a rich media. If it cannot be cultured in a rich media, if it go in our gut system, then probably it will fail. It will not survive there. If it cannot survive, then it cannot translate its beneficial effect to the host. So therefore, we thought that few of the functions of these probiotics are mediated through their surface, and we have a lot of surface protein. It may be the surface protein of these probiotic may be the effector molecules. Then why not to clone these molecules and use them for the same function? So therefore, collagen binding protein, fibronectin binding protein, mucus binding protein, and then S-layer protein. We clone large number of these proteins. So here I'm giving the example of collagen binding protein, which will bind with the collagen and as well as with the mucus. So this was produced, purified by affinity chromatography, and then it was shown to be like a single peak. There is no contamination of any other protein in the SDA space, and this was shown to be authentic protein, that is CBP, by Western blot. And then we wanted to see whether it has the activity or not. If it has no activity, for activity, it means it should bind with the gut epithelial cells. If it cannot bind with the gut epithelial cell, it cannot translate, it cannot immunomodulate, it cannot displace the pathogen. So these are the bright field view of the gut system of the human. So slide was prepared from the tissue, and this is the negative control. We did not use any protein in this one, but here we use our protein and then probe it with the antibodies. So you can see a lot of part of the tissue is bound with our protein, or you can say our protein is binding with the tissue, and very well you can see the features of the gut system, gut tissue here. Here also you can see, so that is the background. But if you wash it a little bit more, you can remove this background as well. And this comes as a nicely fluorescent surfaces. Now you think of, it looks like that your protein is bound all over the surface like a blanket. It means if it is binding to the surface in our gut system, if a pathogen comes, and pathogen also use the same receptor with which these protein are binding, probably pathogen will be excluded, or its transmission or its transience through the gut system will be faster. It will be cleared from the gut system in a faster way as compared to when there was no protein or when there was no probiotics. That is the function. So we did some experiment we did some challenge study in the cell line. So we found that binding of collagen binding protein can dislodge or exclude the binding of pathogens, expedite by 60 to 70% at something like 200 microgram. If you increase to 400 microgram, maybe it can reach to 80%, and that's a big gain. It means we don't have to maintain the probiotic cells, because sometimes if some people are if they consume the probiotics, because they are lactic acid bacteria, they produce the lactic acid, they cause acidity. Also, like this black fungus, which is a opportunistic pathogens, lactic acid bacteria can also become a opportunistic pathogen in immunocompromised subjects. So therefore, we should avoid giving the probiotics to those people. Of course, they have their own advantage as well. They can help making a better immune system. So, but all the probiotics cannot carry out each and every functions. So therefore, we should be cautious about this thing. Rather than this thing, if we produce this protein and give this protein for different targeted function, then we can achieve that function, like fighting against the pathogens, and that has done very well. And DSC, differential scanning calorimetry. So this was done to check whether our protein is like native state, because if it is not folded, then probably it will not denature. So if you increase the temperature, it will go and give an endotherm. And this endotherm, and the area under this endotherm is the enthalpy. It means enthalpy is coming from the contribution like hydrophobic interaction or van der Waals interactions or different kind of interaction which are there, which help then that protein to fold into a three-dimensional structure. So therefore, if it is making a three-dimensional structure, it will give a good endotherm. And a single endotherm means it is folded into a one type of conformation. It means there is not a multiplicity of the conformation. So this is important to know this. Now, I think I'm crossing the time, but I will go a little bit faster. Protein production at factory scale. Usually we go with the yeast, not E. coli, because in E. coli, we use episomal plasmid. That means we can autonomously replicate. But to maintain that plasmid, which carry out the construct, your gene, we have to provide some pressure. Pressure is antibiotic. If we add the antibiotic in the system, only that plasmid will be maintained, because the antibiotic resistance gene is present in the plasmid. The host is sensitive to this resistance gene, antibiotic. So therefore, if we have to maintain the plasmid within the host, we have to always use the antibiotic. And if you are carrying out a fermenter at thousands of liters, then you can calculate the cost of antibiotic which you will be using for maintaining the plasmid. If you don't use the antibiotic in the system, then over the time, the cells will lose the plasmid. Curing will take place. And when cells will lose the plasmid, then what will happen? There will be no production of the protein, because they have already lost the genetic material which is required for production of your protein. So therefore, industry does not take this type of chance. And also you can calculate the cost of the antibiotic at thousands of liters. So it will increase the cost of production of your biopharmaceutical. So therefore, even if it is very simple, but it has some disadvantages. I'm talking about the E. coli. So people go for the yeast. And yeast, we have the Pichia pastoris, which is a similar to Cerevisia, but different group. Similar means its doubling time is very quick, not like E. coli, but approximately 5 to 6 hours. So in a few days, we can accumulate huge amount of biomass. More the biomass, more will be the production. But in this case, rather than having the episomal plasmid, we integrate the plasmid into the genome of Pichia pastoris or yeast, so that when the cell divide, or when the chromosome of the yeast or fungus or anything, they divide or replicate, then our gene present as a part and parcel of the genome will also replicate. It means you don't have to use the antibiotic for the maintenance, because it is a stable integration into the genome. And here is the difference between Cerevisia and pastoris. Why we use pastoris rather than Cerevisia? Because in Cerevisia, in a given time, the amount of cells, biomass produced is only this much. While in pastoris, the amount of biomass produced is huge, almost 50% of the volume is your biomass. And therefore, more the biomass, more will be the expression. And large number of proteins of different origin, like bacterial proteins, yeast protein, plant protein, invertebrate protein, mammalian protein, has been produced in Pichia. It means Pichia is a very good system. And you can produce 22 gram, 2.3 gram, 12 gram, huge amount per liter. And this is a huge amount of protein. In case of E. coli, you can produce like 50 milligram, 100 milligram per liter. But here 22 milligram, but not all protein. The expression depend upon what kind of gene is there. Interferon tau is produced at only 10 milligram, and that's why the cost also depend how much you can produce. If you can produce large amount, cost will decrease. If you are producing very small amount, the cost will increase, production cost. So for the Pichia pastoris, I will skip this one, because here idea is that methanol is a inducer in this case. Like in E. coli, we have IPTG. Here, methanol act as a inducer for the gene expression. So because methanol, which is the simplest carbon source, will be used as a carbon source in the by the yeast when there is no other simple source like glucose or any other sugar. If they are not present, then methanol can be used by this biochemical pathway. So if this is the case, if AOX is produced, it means AX should be produced in large amount. If it has to be produced in large amount, it means its promoter should be very powerful. Its promoter is very powerful, then people thought of why not to replace, why not to clone this promoter in a plasmid here, this green color, and place any gene downstream of this thing. Then when you induce it with methanol, this promoter will produce large number of transcript of any gene which is placed here. So that was the idea. And it is inducible. It means whenever you want to induce, only then production of the protein will take place. So that was the idea. And here is the geosin. Geosin act as a marker protein, or when you are using for transformation, you will differentiate between the transformant and non-transformant, whether this particular plasmid has got integrated into the genome of the host. So there are different methods. They can get integrated into the genome by replacement of some gene like UX gene or his gene, or they can integrate it by insertion. So I will not explain all these things because we don't have the time. So the selection of the host stem for the expression of this protein in yeast. So we can use X33 wild type selection of geosin resistant expression vector. So other are for other type of proto or this histidine dehydrogenase based expression system that I didn't talk about, but we need to remember that we can use X33 for geosin resistance and selection based on geosin. And this is the protocol. You can see just clone the gene, transform Pichia pastoris, then select the transformant and pick 10 to 20 colonies which are appearing on the geosin plate, and then see the expression level, and then choose few of the colonies for overexpression. Then we use this system to produce a Buffalo SLLP1 that is sperm lysozyme-like protein 1. So the sperm lysozyme-like protein are present on the sperm. The name itself suggests. So their structure, their sequence matches with the lysozyme. Then people were wondering what they are doing on the sperm. Lysozyme function is to as antibacterial. It protects us against the pathogens. It is present in the milk. It is present in the saliva. It is present in the all the exodus wherever they are taking place, like in the tears where there are chance of pathogens. So lysozyme is produced. So to lyse the bacteria cell wall. But what the particular lysozyme is doing on the sperm? Then we identified 5 different kind of lysozymes on the surface of buffalo sperm by LC-MS technique. We cloned all these things. We try to understand what is the sequence and structure analysis. What is the relationship between the structure and the sequence, because 5 different isoforms, and they were differing from the lysozyme, which is biologically active. Few of these sperm lysozyme-like protein, they were not active. It means they cannot lyse the bacteria. They cannot hydrolyze the bacteria cell wall. If they cannot hydrolyze the cell wall of bacteria, what they may be doing? So people, if they were thinking that if they are present on the surface of the sperm, maybe that is to protect themselves against the bacteria in case of some infection. But usually there is a blood and barrier barrier between the testis and blood. So chances of infection are lower, but still they are there if there is some wound or some infection takes place directly from external wounds, etc. in the testis. So how to protect themselves? Maybe by this lysozyme-like proteins, but some of these lysozyme-like proteins were inactive. If they were inactive, it means they cannot play a role against the against any infection. If they do not have the role in the protection of the host, then what they were doing, that we were trying to understand. We express in P. pastoris. We also try to produce in E. coli, but that was not successful. So we went to P. pastoris. It was produced 14.4 kilodalton, similar to the lysozyme. Then we tried to check its spectroscopic property. Then it was similar to the lysozyme. It means what we produce in P. pastoris was having a native structure. And then we did some binding study with some another protein. It means if it can bind a protein to another protein which can bind with the lysozyme, it means it has a native lysozyme-like binding cavity. So that we showed it here by the gel retardation assay, and here there was a competitive assay which showed that these protein can bind to a ligand which is binding with the active lysozymes. And here is the fluorescence studies which indicate that there is a quenching of fluorescence when it is binding with the inhibitor. It means by these few studies, we wanted to show that this lysozyme-like protein has a competent ligand binding active site, but they were lacking there was a mutation in the catalytic residue. It means they cannot carry out protective function, but still they were having the binding site with the ligands. It means what they must be doing, because then we took the oocytes, buffalo oocytes, and allowed binding of the sperm lysozyme-like protein with the oocytes. And you can see this ring. This is binding on the zona. And here is the DAPI or PI, this one. And you can see nice fluorescence in the zona region. It means this protein is binding, has a receptor in the zona. If it has a receptor in the zona and it is required by the sperm to interact with the oocyte, it means it may be helping in sperm oocyte binding. If it has a sperm oocyte binding, then it has a bigger role rather than the protection in the reproduction. Like for example, this has the sperm. It has a SLLP, sperm lysozyme protein, and this is the egg or oocyte. in oocyte there is a receptor, and for example, this receptor is for the SLP, so it can bind with this receptor. But if we block this receptor with another protein, another ligand, it means sperm lysozyme like protein cannot bind with this receptor because we have already blocked this one. It means by competitive inhibition, we have blocked this one, and sperm cannot bind, and hence there will be no sperm binding. And we showed it by in vitro fertilization that this is the cleavage in different concentration, like B, C, D, E are different. The blue is control, while this is 50 microgram, this is 100 microgram, this is 150, 300 microgram of SLLP, with which we incubated with the oocytes, and then sperm were inseminated. So this is the decrease in insemination or fertilization, I would say, not insemination but fertilization, and this is the decrease in the morula stage, blastocyst, and H. So they are all going down. So morula and blastocyst formation were going down by 85%. Okay. So we also thought that there may be some NAG because N-acetylglucosamine, it can bind. We did some molecular dynamics and simulation study that they can bind with the Lis5, Lis6, Hagen-White as a control, and then TRIP and these SLLP2. So they are also called lysozyme-like protein or Lis5 or SLLP5, something like this. So in all these things they can bind, but there are two binding mode A, B, C, because there are three NAG3. So one NAG binding here, another binding here, another binding here. So three pockets. So by this one, there are A, B, C, and B, C, D. There are two modes. We showed that probably energy bind in the BCD mode, and this was gain in energy binding by binding this one. And this work was published. So I will skip a little bit because I have already taken a lot of time. So we also filed one patent for composition for modulating fertility. As I explained here, if we can block here this receptor on the egg, these oocytes, then sperm cannot bind. Or think otherwise, if we can block this SLLP present on this sperm, then also it cannot bind with this receptor. We exactly did this one. We took another protein which can bind with the SLLP and incubated this sperm with this lysozyme binding protein, which was an inhibitor, and then used this LBP loaded sperm and carried out IVF. And then there was no fertilization even at 10 microgram of LBP. It means it's highly effective, right? So we filed for this patent for this protein, not for the SLP, but its inhibitor. I will go ahead. So that was all I already explained in the previous, all these things. So here is happy recombinant natively folded proteins. If you are producing a protein, it should fold, it should carry out its function. Just production of protein is not sufficient because most of the time, if the protein needed post-translation modification, then and your activity of the protein depends upon this post-translation modification, like glycosylation for interaction with the receptor or with another protein, or for its stability. Probably protein produced in E. coli may not serve the purpose. Then you have to go to bigger or eukaryotes system, mammalian cells or something like this thing. It should fold, it should be correctly fold, not misfolded, and it should carry out its biological function. So if it is biologically active, you don't have to carry out how it folded and what made it to fold. So therefore, my talk ends here. If you have some questions, I am happy, but I have already closed time limit. So thank you for your patience.
Thank you very much, sir, for your enlightening talk. Sir, I'll just see if there are any questions and revert back. Dr. Ingole, can you see any questions?
Yes,
Dr.
ma'am,
Var-
Dr. Vas. Yeah.
Yeah.
Yes, Dr. Kaushik, it was a very nice and splendid lecture. You throw light on multiples of practical uses of the alternate therapies. We have some questions in chat box. Can I read for you, Dr.
Sure.
Kaushik, if you don't mind? Yeah.
Yeah, please go ahead.
Yeah, sir. What's the practical? It's from Dr. Vasudhara Bhatt. What's the practical problem using picha as vector compared to E. coli?
Easier because it takes 3 to 4 days and it requires the fermenter. If you do in shake flask, then it is very difficult to get sufficient amount of protein even after 3 or 4 days. So that is the biggest problem. And then we have to use the methanol. Methanol is inflammable. So you have to have a control over the feeding and you have to have the continuous feeding of in the fermenter. So therefore you need a lot of investment in the fermenter and then you have to optimize a lot of things because PCA, it produces large amount of the protein, but it is not easy to optimize the condition. You have to optimize the bioprocess to many things like feeding rate, then what are the nutrients, then what is the oxygen rate, because the OD reach could be 300 or 400 OD, while in E. coli usually 1 OD, 2 OD can reach. That is the biomass. But in PCR, biomass is very high. Therefore, you need special fermenter which can rotate at 600 or 500 RPM at very high density of the biomass. And when methanol is fed, it could be toxic. So therefore, you have to optimize at what rate you will feed the methanol so that it doesn't get accumulated into the fermenter. But once you do all this thing, you can get good amount of almost close to native type of proteins.
Yeah, from the same person, there is another query. When the N and C lobe of lactoferrin was separately synthesized, did it affect the biological function or structure?
Yeah, I showed that when N and C lobe were separated from each other, because, for example, if this is the structure of lactoferrin, like N lobe and C lobe, and there is a hinge region there, and this hinge region bring them together, and there are interdomain interaction. This interdomain interaction affect the binding and release of iron as well as the stability that we checked. So when you separate them, then their stability decreased. When they were hollow protein, then their stability was higher. As well as we wanted to see whether it is the N lobe or C lobe which was binding strongly with the iron. N lobe is binding more strongly. Now C lobe was binding more strongly than the N lobe. So N lobe function, the binding decrease as compared to when it is in the hollow protein. When N lobe and C lobe were together, then N lobe was releasing or binding iron more strongly. But when N lobe was separated, then the binding strength was decreased. But that also happens not with the recombinant molecule, but when you separate N and C lobe from the lactoferrin present in the milk. So they were also similar.
Okay, thank you, sir. I hope Dr. Bhatt has got the answer. Another query is, can we use the human or animal gut cells for transfection purpose after selection markers? For example, using the rabbit polyclonal antibody, can it be applied for those cells?
I couldn't get. I think I have a lot of birds outside. They are making a lot of noise. So can you just repeat it?
Yeah, sure. Can we use the human or animal gut cells for transfection purpose after selection markers? Say, for example, a rabbit polyclonal antibody is applied, if can be applied for those cells.
I am unable to listen to your first part. Sorry.
Can we use the human or animal gut cells for transfection purpose?
Yeah, sure. Why not?
Okay.
Yeah, they can be used.
Yeah, right. Then another one is, do you examine the receptor of REC-SLP1 on zona-free oocytes and development stages like the 2-cell stage?
Yes,
From
we
your
did
colleague
it.
only. Okay,
Yes,
sir.
we did
Yeah.
it. We removed the
Right,
zona,
sir.
and so this protein didn't bind.
Okay, the one more which is coming up is if we express any protein prokaryotic system, then how we know that it is properly folded and do their normal function?
Uh, First thing is, if it is biologically active, straight away check for the biological activity. If it is biologically active, it means it is folded. If it is not biologically active, then there you have to see the circular dichroism spectra, fluorescence, etc., whether they're misfolded or unfolded completely. So if they are not folded, then you have to denature them completely and then refold back them. That is by controlled in vitro conditions. You can try to find out the condition for refolding. That also we do sometime.
Yeah, thank you, sir. And I hope running with the time, we have the last question. Did you try expressing lactoferrin in E. coli system?
Yeah, we did
And
it.
what are the results? Okay, sir.
Yeah, we did it, and we could, but it was not into the proper three-dimensional structure.
Okay, sir. I think it was the last question what I can see in the chat box. Thank you, sir. Thank you for a very nice presentation, in-depth presentation on the recombinant expression of the genes. Over to Dr. Maji, madam, please.
Thank you very much, sir, for your very extensive and informative lecture. Especially the one on recombinant collagen binding protein was extremely very, very informative to us as veterinarians, because we are always trying to use probiotics in poultry and other animals. Sir, is there any idea whether this recombinant collagen binding protein will enter the market in the future, sir?
Idea was that, but we had to carry out the safety study and all these things before they come. We had to carry out the mice study also, in vivo studies. So only after that, we have carried out in only cell lines so far.
Okay, so it's very promising, and we hope that definitely it will see the light of the day. And also your work on the recombinant strains of Pichia pastoris was very enlightening to all of us. I thank you very much, sir, for taking out your time and delivering such an extensive and informative lecture to all the audience. Thank you very much, sir.
Thank you very much. Thank you. And thank you to giving the opportunity to explain our work. But I try to make it like so that people who are not in this technology, they can also understand it. So I started with the vectors, promoters, all those things. So
Yes,
that
yes.
I was informed that probably some of the participants may not be aware about those things.
Yes, sir. It was very nice. I really enjoyed it. Thank you so much, sir.
Okay, thank you. And sorry that I took a little bit extra time,
No, it's
but
okay. No, it's
my
fine.
time was taken by Naresh.
Thank you, sir.
Okay, bye-bye.
Bye.
I can see Ram. So you have next class, Ramesh, Naresh?
Fine, sir.
Okay.
Yeah.
Okay, bye. I'm signing out.
Thank you, sir.
Okay, bye-bye. Bye.
Sir, it
Thank
is
you, sir. Thank you so much.
question answer over.
Before moving to the next lecture again, I would just like to announce that.
Sonal, ek minute.
Yes, sir.
I would like to thank Dr. Kaushik sir. That is why I interpreted. Sorry for that. Kaushik sir, thank you so much for accepting our invitation within very short time, and you have delivered very nice and very informative lecture. Thank you so much. Okay, now you can continue. Sonal.
Yes, sir. So before moving on to the next lecture, I would just like to announce that after this last lecture of this training, we will be having the concluding session. So all the participants are requested to please stay with us even after the lecture is completed. And again, the next speaker with us today for this last session is Dr. Naresh Selokar again. And now he will be discussing about the ethics involved in animal and gene cloning. So again, I request Maji ma'am and Vaska sir to please take over.
Good afternoon to you, sir. Dr. Selokar, I welcome once again for this next session. and we look forward to your lecture. So without any delay, so please start.
Thank you. Thank you, Dr. Manji ma'am. Slides are visible.
Yes, sir. No, yes, they are visible, sir.
To make it a big screen. Now it's visible now? Right?
Yes, yes, yes. They are
Yes,
visible.
sir.
Okay.
Then bolo.
Yeah. Now this, I think, the two talks which I was delivered earlier, it was the animal cloning, gene cloning or gene editing. And recently the excellent talk is given by Kaushik. He has been talked about how this gene cloning, particularly expression of recombinant proteins. So in particularly in E. coli system or in yeast, Pichia, or the expression of what the recombinant protein he has been expressed and their functionality, how they have been modulated. It's one. So, and before that also the many speakers, they have been talked about is all the kind of these gene, including the cloning technology, gene cloning or the expression of recombinant proteins or the therapeutic proteins or antibiotic production. So now I think is before the closing of this training section, I think every attendees need to be aware what kind of the regulation we need to follow or what is legally allowed. So it is not every work as legally allowed. And you need to follow the guidelines, how you, if before initiating these gene technology work, which include the gene or the pathogenic words, even the bacterial pathogens, pathogenic bacteria, you need to follow. So I will try to give an information about the available the guidelines and what is the currently available of the genetically modified organisms, because I will morely focus on the genetically modified the organisms, including crops and the plants. And then we'll understand, I will give you some information about which are the plants particularly and the animal products have been commercially available for the human consumption. In addition to that, there are many proteins, drugs, and vaccines. They have been produced through the recombinant DNA technology in vitro or even in vivo. They have been already in use. For example, the human insulin is one of them, or even for example, the vaccines, like the COVID vaccine. We are the vaccines from these mRNA vaccines, attenuated virus vaccines, so from also, or the recombinant subunit vaccines also available. But I will not go into much detail about the recombinant protein expressed in vitro, but I will just give you more information, stress more related to the genetically modified the crops and the plant, the crops and the animals. Now coming into the what is the gene cloning is. So gene cloning is the meaning of the cloning. If meaning of the cloning is a word is coming from the Greek, is a twig. So what is actually the cloning? I will just go into a little bit deep introduction part, and then we'll go to the regulatory issues. So cloning is asexually reproduction or production. Asexual production means there is no unition of the male and female gametes. For example, the replica of the DNA, antibody, mRNA, organism, cells, bacteria, anything that is called the multiplication, that is asexually multiplication of any micromolecules, molecules, or the organism, including the animal. So if we talked about the individuals grown from the single cell, for example, the cloning, cell cloning, in which the single cell need to be placed in a culture, so more population of the cells will be generated. That is also called as the cloning. That is called the cell cloning. And the important thing is to keep in mind that they all are the genetically identical, or their sequence. For example, the antibodies, they are the structure is 100% identical, or the DNA, the sequence is 100% identical. It isn't worked on the cloning. Now coming into the types of the cloning, what are the types of the cloning? I think many the speakers before me, they have been talked about the different types of the cloning, gene cloning. I think recently Dr. Kaushik has been well explained about the gene cloning, about how the DNA fragment is need to be inserted into the vector. There are the different vectors. He has been talked about the shuttle vectors, E. coli vectors, the yeast vectors, or the combination of vectors and the promoters. I will not be into. And then we have the recombinant DNA, and this recombinant DNA will either express the protein for example, the recombinant that is the lysozyme he has been expressed in his laboratory, or the recombinant human insulin. The second cloning method which has been used in the cell cloning or somatic cell cloning. The cloning is particularly in case of the crops and the plants is well established, particularly in crops. There are many varieties or the varieties of the plant has been developed, in which what happened in the plant as a character is brought from the single cell this apple to generate the entire plant. And this is called the cell or somatic cell cloning. For example, in case of the animal, the single cell clonal proliferation will be generated in the plant. That is called this somatic cell cloning, or the many varieties of the plants. In bacteria, for example, transformation from single bacteria, so bacteria is formed. It's also a kind of the cloning from the single bacteria. If they are transfected with this any kind of recombinant DNA protein, all the bacterial cells, they have the same copy of the recombinant DNA. Now, the third important aspect of the cloning, particularly for the production side, is the animal cloning, which I have been talking my last the talk. I think Dolly, I think everybody's been the Dolly, everybody's aware that the Dolly is the clone ship has been first clone ship is produced from the differentiated somatic cell. People, they have been confused that Dolly is the first clone. If you ask if Dolly is the first clone, no, Dolly is not a first clone. It is a first clone from the use of the differentiated somatic cell. Before the Dolly, there are the clones like the Megan, Morag, or any other several clones has been produced. But in that case, the previous to the Dolly, the somatics, the donor cell are used as the embryonic cell. That is the embryonic cell, like the blastomere. What the Dolly has been changed, the perception is that previously, like the central dogma of the molecular biology, that the DNA will transcribe into the RNA and the RNA will translate into protein. Similarly, in developmental biology or embryology, it is believed that the embryo will develop into the germ cells or the germ lines, and then from the germ lines, there is the differentiation of the cells like the ectoderm, endoderm, and mesoderm, and the body organ form. And there is not a possible that any differentiated cells will be read back into the embryo state. But the birth of the Dolly has been disprove the concept of the central dogma of the developmental biology. Now it is possible that any kind of the cell, differentiated cells, either the skin cells or the hair cell or muscle cell or intestine cell, there has been potential to read back into the totipotent cell, that is an embryonic cell. Also, when the invention of the induced pluripotent stem cell emerged out, for example, the Yamanaka and Takahashi, they have developed the induced pluripotent stem cell. That also been challenged to the central dogma of the developmental biology. That against these differentiated cell read back into the totipotent, there is a multipotent stem cells. Anyway, this is also a cloning, animal cloning, in which the multiplying of an animal. Suppose you want an animal, take a cells and multiply this animal. And the fourth important aspects of the cloning is the therapeutic cloning. What is a therapeutic cloning actually is particularly for the human application. So what is actually allowed in therapeutic cloning? So it is through the therapeutic cloning, it is impossible to multiply the cells, either to replace the damaged cells in the human or to correct the disease. There is a stem cell therapy, and people now been working on to replace these the cancerous cell, blood cancerous cells with the stem cell that has been have the potential to correct the blood cancer. Also, there's somebody have the damage in the liver. The people are the clinical trial has been ongoing to transplant the stem cell into the liver so that the damaged cells need to be replaced. So this is these all the type of the clonings they have been available, and I think those have been hardcore molecular biology, particularly for recombinant expression.
they are going to use the gene cloning. Suppose in the plant science or even from the animal science vectors, they use the somatic cell cloning. The reproductive biotechnologies, they have been working on the animal cloning, and the human application, the researchers, they are working on the human, they have been exploring the therapeutic cloning. Now, I think I will not go into detail because I have been talked much in my first talk why we need these, all the kind of technology. Just highlighting is that in 2050, the global population expected to 10 billion. And this 10 billion, because we have the limited land, we have the limited resources in terms of the use of the water, in terms of the availability of the energy. So within that limited resources, we need to feed the people of the 110 billion population. And this 110 billion population, when the population increases, we have the deforestation. There is a climate change is significantly affecting the everywhere. We also experience the change in the tremendous change in the climate change is there. So in addition to that, we wanted to what happened in 2050, the more than the 60% of the carbon footprint should be the less our target. So how we can achieve? Because if we talked about the Indian condition, we know we have the huge population, I think 190 millions of cattle, and total domestic animal population, more than the 500 millions of the domestic animal in India. But despite that, our productivity is the less. So we have been maintaining these animals. They will require the feed, they will require housing, they will raise the grazing lands. But in addition to that, what our target was to decrease the population of the unproductive or low producing animals and to have more population of the productive animals. Similarly, in case of the crops, because in crops, the day by day, the land holding intensity is reduced because of the urbanization or the developments. So within that land holding of the land, whatever is available for agricultural application, to increase the productivity of the crops or the vegetables so that we can feed the population. With considering that also, we also need to protect the health. I think COVID is one of the examples to maybe think about the direction. So my aim of this today's talk to highlight each and every aspect, not necessarily to work on any aspect to anything that can be modified, that can be produced, like the production of monster animal or the production of the vegetables. But we need to think when we use this technology very cautiously. And the caution is to keep in mind to not only if you are aiming to improve the productivity, but also aiming is that how sustainable the technology will be for the future. So I will try to highlight some of the aspect of this. Also, while working on the gene technology, we need to follow the regulatory guideline, because if you are not been taken proper care, maybe because of the lack of information with you, or you are unintentionally has been done, so then there will be the serious consequences. So we need to keep in mind, we need to take an approval from the regulatory agency too before initiating this work. Now the GMO, if we talked about the genetically modified organisms, so I will not go into detail what are the genetically modified organisms that, but when the GMO has been started, it has been evolved and evolving. There are many crops that have been produced, the major crops which we are being currently eating. So don't think is that you are not eating the genetically modified, the products from not exactly the genetically modified organism, but the product. For example, soybean oil, or example, cotton. The cotton is, or the BT cotton is a well-known example of the genetically modified, the crops of the plant have been used in the country. And similarly, in example, the corns. So these are the three major, the crops has been produced. Now, India, if you talk about the India in the last year, 2018, there has been debate, or the bill has been placed in the parliament to release genetically modified mustard, which has been developed by the Indian Council of Agriculture Research, Department of Biotechnology, Delhi University. They have been developed this genetically modified mustard. These have been developed almost 20 years before. A lot of the clinical trial has been done, but still again government has been not approved. But in spite of that fact, what the canola oil, I think you maybe heard about the canola oil, which the Indian government has been importing from the Canada. This canola oil has been produced from the canola. This is a rapeseed, which is also genetically modified. So in India also, we are being consuming the products from genetically modified organism or the plants or the animals. Similarly, for example, in Bangladesh, our near country, which has been approved the BT brinjal. In India, BT brinjal already has been produced and developed, but Indian government not in India. There is only the crops which has been allowed to cultivate in the in agricultural field is BT cotton. Rest of these one they are not allowed legally. So we need to also keep in mind. But what I would like to highlight is that so because in the Bangladesh they have been allowed to cultivate the BT brinjal, there might be chances that the BT brinjal that has been developed in the Bangladesh may import some of the neighboring the region of the Bangladesh will import and they will have consumed the BT brinjal. Anyway, these are the plants, animals, and what are the agencies in the United States or in European countries or in the Asian countries? What is their opinion is a talk about? If we talked about which country is the largest producer of genetically modified organism, when I was talking about the genetically modified organism, mainly focusing upon the crops and animals, is not the microorganisms like the bacteria, yeast, or something like the recombinant DNA technology. So the USA is the largest producer of the genetically modified organism. So and apart from that, which country they have been not considered that the genetically modified organism are safe for the human consumption and not legally allowed to cultivate any genetically modified. One is the European countries. The majority of European countries, they have been banned on the genetically modified. Australia and the New Zealand, they have been banned on the genetically application or the cultivation of the genetically modified organism. Now coming to the GMO in animals, if we talked about the GMO in animals, in animal only the salmon. The salmon is a fish which has been developed by in Canada and USA, one commercial company, biotech company. They have been produce this genetically modified salmon, which is approved for the human consumption. In spite of that, this approval takes almost more than 13 years to reach to the human consumption. Now it is approved. In 2017, it is approved. But other genetically modified animal the products, if we talked about, there's Atiran is a well-known. There's a human anti-thrombin. There's a clotting factors, so act as a anti-blood clotting agents, which has a deficiency of the thrombin. So Atiran has been produced in the goat. The human beta-cholecystectin protexia is also been used as a down the toxin, which has been working on the nerves, which has a neurotoxicity, which has been, but this is already been approved. This is under the clinical trial. Similar one is the clotting factor 8. It's called the Elocate, which has been produced in the pig, which is particularly for those patients which has the hemophilia. I think hemophilia is also a genetic disorder in which if anybody has now the continuation of the bleeding, so that's not been stopped. So to that, this is the recombinant protein as expressed in the pig. So and another one is this one, is a human fibrinogen or human C1 inhibitor, the esterase inhibitor, the Roconase. This has also been approved recently in 2017 or 18 for the human application. This is again a genetic disorder called angioedema. The angioedema is a skin disorder, allergy. Some of the people in the European ancestry, they have this disease, and Roconase has been legally approved now for the human use in USA and European countries and all over the world. The human myelin basic protein is expressed in the cow. There is a multiple sclerosis. I think people, you maybe have heard about the multiple sclerosis, and this has been developed in New Zealand. AG Research, this is Agricultural Animal Research Institute, and this is again in the research development stage. So what currently we have for the application of the GMO products, particularly that they produce in the live animal, these are not produced in vitro, they are produced in the animals itself. All these are the transgenic animals, and this is one is the Atiran, Protexia, Ecolate, and the fibrinogen, Roconase, and the myelin. These are a couple of, out of them, the 2 or 3 proteins only, they have been approved for the human use for the treatment of the disease. And so for the human consumption, as I told you, the Bounty salmon. Now coming into the difference between, I think many people now, because I have been talked about the gene editing, the people, they have been confused about that they are a genetically modified organism or the gene edited organism or animals are the same. No, there is a significant difference. Just I would like to take a couple of minutes on this, how to explain about the techniques, how the technically difficult. The GMO, if we talk about the GMO in which the foreign gene means, for example, we wanted to express, example, the clotting factor, like the fibrinogen we wanted to express into the goat. So in that case, the fibrinogen of the human will insert into, for example, this is a fibrinogen of the human, will insert into the goat genome, and then they will express in a blood. So, and from the blood, from the plasma, it will be extracted. This is for the genetically modified organism, or for the anything. If you wanted to express anything, insulin or anything, they will need to be expressed in the milk or anything. That is called the insertion is very important. If you insert the foreign gene into the genome of any organism, that according to definition is called the genetically modified organism. But when the CRISPR has been evolved, it is possible is that only it is possible to cut the DNA and modify it. If you wanted to change the one nucleotide or two nucleotide, I'm giving you, I have just explained my last example of the knocking out of the myostatin. So knocking out means there is a changing of one or two nucleotide. In this case, particularly gene editing, there is a no insertion of any foreign DNA. And these products, now the regulatory agencies, particularly in USA, in Argentina and Brazil, they have been approving this the gene editing technology. They will not consider in this gene editing product, which are the small deletions or small insertions, as a genetically modified organism in which the large chunk of the DNA, for example, foreign gene is inserted into the genome. But if you look at the characteristics, what happened in the phenotype of the animal, in the phenotype of the animal or the crops, 100% phenotype will be changed in case of the GMO. But in case of these gene editing animal, there is a not significant change. And if you look at the genome level, it will really indisputable change in the genome. And this technique is considered to be similar. And the scientists who are working on the gene editing, they're arguing that we need to think is that the gene editing technique to be similar as the traditional breeding technique, for example, or a farm animal breeding technique, or in crops, that is site-directed mutagenesis has been doing for many years in a crop using the site-directed mutagenesis. Similarly, there are in the still now no crops has been approved for the human consumption through the gene editing. They have been conducted for the clinical trials. We need to conduct the clinical trial phase 1, phase 2, and phase 3, and then only they are going to approve. But some of the products, I will go by slowly. So, what are their opinions of the different countries on the regulation of the GMOs and the gene editing? Now, what are the pros of the, if you look at the pros of the genomic modified organism? So, as I told you, I think just quickly summarizing what are the benefits of this. The benefit is that it can increase the crop yield, animal yield, or anything in. It contains the higher amount of beneficial substances. I think you people have been heard about the golden rice. I think if you talk about in India, we have the deficiency of vitamin E in childs, and because of that, many childs are the blind. To address this issue, the scientists have been working on to develop rice variety which has the higher content of the vitamin A, and they have developed that rice, and that rice has been called as the golden rice. It has not yet been approved for the human consumption, but that rice has been developed because in any kind of the genetically modified organism, it takes an years to study their efficacy, their health effect, what their effect on environment, and then only it been regularly approved for the human consumption. This is one of the examples. To make the plants more resistant, there are the couple of the plants. For example, in India, the people are working on the banana, which has been resistant to the virus infection. So that this plant is more resistant, or to also increase the shelf-life of an particular the products or the meat. If you talked about the crop, the product like the tomato, you know that the tomato has some days shelf life. After 5 days or 6 days, the tomato cannot be used. They have been rotten. So scientists have been trying to improve the shelf life from weeks to the months, and they have been succeed, but still we need to take an approval. Also, the efficient use of the land, efficient use of the food price. If you have more production, healthier, it will be obviously it will be reduce the land use. That is our ultimate aim. And if we produce the more productivity, the less manpower need to be focused on the agricultural work. So also, if we have need to use the now for example, the BT cotton is one example. So in BT cotton, before the BT cotton, I will not show in the graph how the BT cotton production in India. Still today, 70 to 80% cotton grown in India are the BT cotton, because there is a bulbwood, is a worm is there, they will infect the cotton and then yield is being less. So there is all the birds of the cotton will be destroyed. In BT cotton, there is the gene which has been injected, inject by the insect, and they will produce the toxins in the guts of the insect and vector, that is the fly or the insect has been died. So with the use of the genetically modified organism, we will reduce the amount of pesticides in case of the crops. Also, will reduce the amount of the antibiotics which are being using for the treatment of the animals. For example, if the animal succumbs to the infection, then we go for the antibiotic treatments. That will also reduce if the animal resistance to disease. Also, the use of the antibiotics or the probiotics for the particularly for the poultry and fishes. In poultry industry and fish industry, the majority of the amount of investment the people or the poultry rearer will invest on the amount of the feed, probiotics and antibiotics or amount of feed. If they have been modified itself, is that the productivity is increased, then the food consumption of the poultry or fishes are less. Also, there is a less amount of antibiotics will be used in the poultry or the fishes. Ultimate aim was to have does not have the antibiotic resistance as human. These are the some of the pros of the GMOs. If you talk about the what are the cons, so the cons is that I think there has been debates from years and years. Is that GMO is not good? It is a bad for the human. There are also religious concerns, like in Europe, the European people, they believe that the use of GMO has been hampering to environment as well as religious sentiments to them. So they have been not been allowed. There has been concern. That is the one of the problem with the application of GMO at the larger scale in the world. Also, there has been always been concern the spread of the serious epidemics and pandemics. So, for example, there is an example. There has been hypothesis or speculation regarding the emergence of the COVID. This is the coronavirus, because the people have also been worked on this, the microorganisms, and they have been genetically modified. There is a gene to increase their pathogenicity, or for their research study, they are modified. By chance, there is any leakage of the virus from the out of the laboratory, like BSL-3 and BSL-4 laboratories are required. If they have been infected or leakage out of the, or somebody is infected with the virus, and that is a high spreading efficiency, and that will be significantly impact the global health. And that is one is this is the COVID-19. Also, there is a safety issues, because some of the studies have been shows that the consumption of the genetically modified crops, for example, potatoes or the corns, there have been some changes into the pathogenicity been observed in some of the model animals, like these studies in the rats or the rabbits that shows is that. Also, it is our conception, meaning these are ethicists, they are believing that we are not allowed to any change in the DNA of any either it is a plant or it is an animal or it is a microorganism, because they believe that the God has been created the earth, and the human being is not allowed to do any alteration in case of the DNA, and that has been because of all these the concerns, particularly beliefs of the people or the societies, that the progress of the GMO still has been very slow. Also, some of the studies shows that the allergic reaction, these some of the more these vulnerable diseases have been seen. Also, it has been fearing that if we have been used these, for example, modified these microorganisms or viruses, then there is a possibility that these viruses, because they are emerging out in environment, or they have been emerging out into the host, for example, infected animals or human, and then there will be a resistance, more resistance, in spite of the low antibiotic resistance or antiviral resistance. So all this the factors is being considered to have the impact and the low considered to have the reduced the progress in terms of the genetically modified organism. Also, if you think about how old the GMO technology is, so if you talk about the GMO technology has been 1960s, not more than the 50 or 60-year-old. So people are believing we need to work more on this aspect, need to spend more time, and this technology not yet mature. So for the translation, because of the shorter time that the technology is evolved, so we do not have the long-term consequences. So what will be the effect of the cultivation of the BT cotton after the 500 years? We don't have that long-term consequences data with us, or the 60, 70 years of the clinical trial data with us, and that's is unclear to us. And that all these parameters which I've been talking about will ultimately affecting the growth or application of the genetically modified animal for the human consumption. What is ethically allowed? If you talked about what ethically allowed in case of the production, in any, if you talked about the plant, any gene can be changed for the, if you wanted to study. So any gene for the in vitro study or some of the field trials, it is possible you can allow to change anything. In case of the animals, animals means the laboratory animal, mice, rat, rabbits, farm animals, cows, buffaloes, sheep, fishes, poultry, any animals from the ants. There's any gene can be allowed legally allowed to study and to produce the model animals. But if we talked about the human application, in human the study is approved for the in vitro application only. Suppose for the human cell line, you can do the generation of the transgenic cell lines. There are transgenic cell lines or some gene editing, some recombinant protein studies, expression on the in vitro studies can be allowed. But if you wanted to use for the in vivo, is not been allowed. So in human, there's in vivo study. Suppose somebody wanted to produce the transgenic human being. Suppose they wanted to produce such a human being that this human being will resist to the tuberculosis. For example, that person will never been diagnosed with the disease for the tuberculosis. This is not legally allowed. Suppose somebody wanted to express something, for example, to condition for the baldness. So they wanted to express the gene which is responsible for baldness, to knocking out that gene and express this gene into human and to produce. That is not possible. So also direct application of these infectious agents, for example, the infectious bacteria or the infectious the viruses developed through the application by the human, they are not legally allowed. So if you want to, what is the currently global use? The globally what we have been using the several drugs, vaccines like the COVID vaccines, proteins, recombinant proteins, insulins or other things we have been using, but all these the drugs and proteins, they have been produced in vitro. Some of them are produced in vivo, like the Tyran or the Loconis, they are produced in vivo in case of the sheep and the goats and rabbits. But majority of what are the drugs, vaccine, and the proteins which have been currently using for the human application, they have been produced in vitro, particularly for the recombinant DNA technology, either in yeast or the cells. But regarding the crops, what are these legally grown? So majority of the soybean, if we talked about the 11 countries, they are growing the soybean, which is genetically modified. Similarly, cotton, India is also one of them country to produce, and 80% of global planting of the cotton, they have been grown by the BT cotton. The maize also, the maize 17 countries. In canola, 4 countries, that was I was talking, then the Indian, the scientists from the India planning to approve for the GM mustard for the cultivation in India. So it will not been allowed. But still the 4 countries where the India is one of the importer of the canola oil from the Canada, where they have been legally allowed to cultivate the canola. So, and these are the 4 countries, and the 30% global planting has been shared by the canola. What if we talked about the animal product, which has been allowed for the human consumption, is the salmon fish. When the salmon fish has been begin development, is a journey. This is a company in the USA and the Canada. Biology 45. This is a company, biotechnology company. So the salmon fish is a variety of the fish which is a more growth and they have more meat production in this fish. As then began in 1989. So first has been developed, they have been established to cell line, they produced it, and they have been first applied to submit their application because FDA is a Food and Drug Administration in the USA. It has been regulating the animal product. The USDA has been regulating the crops product, but the FDA, Food and Drug Administration, has been regulating in 2003, almost 2003, is more than almost 20 years before. They place their application that this need to be approved for the human consumption, and through the years and years, in 2017, it has been approved for the human consumption and the production, and now it has been sold in the United States market or in the Canada. So this product has been used. If anybody visited to the countries, they may have chances to use these transgenic salmon fish. Now coming to the now the journey of the GMO, what's the average GMO? Is the genetically, if we talked about the genetically modified crops or the plant, it takes almost 10 to 12 years and millions of the rupees to be before coming to the market. And how many it will take? Almost 15 to 20 years. The studies, if somebody asks to have more studies on the environment, it will take 20, 25 years. And the regulatory process itself, because if wanted to apply for to release this commercially, it will take at least 5 to 7 years. So it's not so easy for people thinking that today we will start working on the production of the genetically modified the organism, and then we will use this organism tomorrow or maybe next year. It is not possible. If you look at how many regulatory countries, if you total countries where the regulatory authorities are available. In India, the Ministry of Environmental Forest, one of them, Department of Biotechnology, Indian Council of Agricultural Research, and this one is ICMR. These are the regulatory agency bodies in the country. According to their expertise area, they have been dealing, like the agriculture application, ICAR and Department of Biotechnology is dealing. Even the Ministry of Environmental Forest, regarding the human health, ICMR is dealing. So it's total 72 countries, they have been regulatory agencies all over the world, including India too. Now the status of the gene editing. If you look at the gene editing is a different, once again, I would like to again stress is that the gene editing is different. And what is because gene editing initiated recently, there are very few countries, they have regulation and they have something, some guidelines with them. If we talked about which countries they have been, do not regulate G varieties with the no foreign gene integration. If there is a no foreign gene integration, these countries, majority, the United States, Canada, Brazil, Argentina, and Chile, and Australia, and even the Japan. These 6-7 countries, if there is a no foreign gene integration, for example, the knocking out of the myostatin gene, then in this situation, these will be no regulated as a genetically engineered animals. Or if there is any crop, if there is a no foreign gene insertion, if only change of the couple of nucleotide in the DNA, then this will be not. Then what is the situation in India? In India, this is a recent, the study has been published in a plant journal. So what they have been shown in India, we do not have yet any regulatory guideline for gene editing. We have the guideline for the transgenic crops and animals, but we do not have any guideline for the gene editing. In 2020, the Department of Biotechnology has proposed a draft proposal, which is available in the Department of Biotechnology website. In 2020, they proposed a draft to formulate a guideline. And I will just share this draft proposal also. Similarly to the India, Kenya and Nigeria also, they do not have, though they are these countries have been extensively using the gene editing technologies to produce the crops and animals. They do not have the much stringent regulation or guideline, or do not have any rules for the gene editing technique. Now coming into the country, particularly European Union, they completely they have been considering that even in the change of the single nucleotide, will treat it as a GMO. That means like the insertion of the foreign gene, and need to be the follow same regulatory guidelines that is following for the transgenic animal. So in Europe, the transgenic animals, including the human animal, are treated same. In case of the USA, Brazil, Argentina, Chile, Canada, Australia, and Japan, the genome-edited animals in which there is no foreign gene insertion need not to follow a GE guideline, that is a genetically engineered or transgenic animal guideline. We India, we do not have streamline. But according to the 20 January 2020 Department of Budget proposed, in this we are following the guideline of the USA, Brazil, Argentina, Chile, Canada, Australia, and Japan. So if this guideline has been approved, then our map will be the dark blue. That means we will follow the guidelines similar to the USA and Australia, in which the genetically edited, genome-edited animals in which there is no foreign insertion will not be follow a regulatory guideline as a transgenic animal. This is there. Now coming into the human, I think the human, it's a totally different. In crops and animals, there are many things are allowed. In human, there is so many difficulties, and is not been. There's a regulatory agency is very, very strict. They have not been allowed. If you look at it, in the majority of countries, the complete ban of any kind of the gene editing works in human, even in case of the cell lines, or even particularly for the embryos, germline editing is completely banned, and is a criminal offense. If you have been attempted, you will be under the bar. So this is, but regarding the against, I was regarding the India. If you talked about the India, we little bit have been developing guideline because the draft has been proposed by the in 2020 by Department of Biotechnology, and according to draft again, it will be a similar to a restrictive type. A restrictive type means what? What is the restrictive type of the regulation is that you are allowed to conduct research studies. Suppose somebody wanted to show that through the gene editing, the sickle cell anemia can be cured, is absolutely fine until the cell stage. But even if you wanted to embryos, the embryos need not to be the culture more than 14 days. So that is a policy of the United States. What is in the United States restrictive means they are allowing some of the research, and there is a debate is that suppose those person has been suffering from sickle cell anemia or Down syndrome, how they are feeling, how the difficulty for them to survive, and according to their views, if any technology will apply them to save their offspring or their child in the future, there may be possible to use this gene editing technology for the human application. But till now, absolutely, majority of countries there is a complete ban of the use of the gene editing or the gene editing, transgenic, CRISPR, or anything. They are completely banned in the majority of the country. Regarding the India and China, we are being little bit we have the ban, but that need to be follow a guideline. Legislation means a complete ban. For example, Canada, Australia, European countries, or in Brazil, there is a complete ban. You cannot do anything. There is no permission at all to even for the research experiment. That is for the legislatively. Ban and guideline means you need to have allow something with the approval of the regulatory authority, and restrictive means use something according to the case-by-case basis. Now coming into because of the many of the people who are attending this one, they wanted to work on this either recombinant DNA technology or the pathogenic bacteria, viruses, or the gene cloning, animal cloning, transgenic. So this is what is the guideline available in the India. So guideline, particularly this recombinant DNA technology, gene technology, is governed by the Department of Biotechnology, and there is a different committees is there. So this is the proposed. There is a publication from the Department of Biotechnology. IBSC handbooks is there. So I urge all these attendees to go through this guideline if you are really interested to work on the pathogenic bacteria, virus, or the gene technology. This handbook, they have been showing all the regulatory frameworks, regulatory the committees, how to submit an application, their formats is there, and you need to follow this guideline. If any case by chance, if you're not following these regulatory agencies and something happened because of your experiment, then there will be the legal strong consequences. So I will just quickly, this is now previously the IBSCs or the all regulatory agencies or regulatory, the proposals, they have been sent by the hard copies, but from last 2 or 3 years, there is a portal has been developed by Department of Biotechnology that is called the IBKP portal. So IB, if you type on the Google, you search on the Google, you go through it. I think each institute, either it's any veterinary college, agriculture university, or the research institution or university, every institute must have Institute Biosafety Committee. And this institution Biosafety Committee will regulate all the kind of the gene technology work. If you're working on the pathogenic bacteria, you need to take the approval from the IBSC. If you wanted to work on the production of the recombinant protein, you need to take the approval from the IBSC first. If you're working on the gene cloning or animal genome editing or transgenic animal, you also need to take an approval for your the studies. Even for the research thesis application, because in NDRI we follow whatever the student they have been worked on this kind of the work, they also their thesis also need to be approved before initiation of implement with the IBSC. And according to experiment, we need to follow the guidelines like the IBSC. Then if your application need to be sent to the RCGM, is not, it will determine by the IBSC according to the pathogenicity of your work. And if the RCGM, if you have developed any product or anything wanted to do the clinical trial, then GEC committee is there. So these are the statutory committees. These are two committees at the institution level, particularly, for example, Bombay Veterinary College, they must have the institution biosafety committee. There is one chairperson, member secretary, members, medical experts. There are a couple of people are there. I think I will not go into detail, but those are interested, they must follow the IBSC handbook guideline 2017 draft, that handbook. The recombinant DNA advisory committee, its function is advisory tribe. It will give the advisory. Advisory means how to, what kind of the facilities you need to have, like the BSL-1 facility, BSL-2 facility, for which kind of the bacteria or kind of the virus or kind of work. It is an administrative agency, is the Department of Biotechnology. Then the second approval committee, IBSC is also approval committee. Another committee is the review committee on genetic manipulation. There is the RCGM, review committee on the genetic manipulation. RCGM, it is a statutory committee at the Department of Biotechnology. And now, because of this digitalization, every 15 days, there's a meeting of the IBSC to consider application forwarded by RCGM. RCGM, every 15 days, there is a meeting. So if anybody have the application fall into RCGM category, they need to be forwarded through the IBSC, and it is an approval authority. The fourth committee is the genetic GEAC, the Genetic Engineering Appraisal Committee. It is a committee, high-end committee or the high-level committee at the Ministry of Environment and Forest. This is ultimate, the committee which is approved for the any clinical or the field trial. It's an approval committee. Another committee is if you wanted to use, for example, I have developed these animals, suppose for example, any transgenic animal wanted to use the field trial in Haryana. Then there is a state government, every state governments have the monitoring committee, and like for the Maharashtra will also have the monitoring committee, Jammu Kashmir, all the state, whatever the state or the territories is there in India, they have, the state government have the state biotechnology coordination committee. This is a monitoring committee. Monitoring committee means what? Suppose you have developed something and you wanted to test that product or the technology into the field, then their aim was to monitor it. Again, there is in every state, they have the district level committee. Suppose I have chosen for example, Karnal district for my field of study. Then each district, the DC is there in district collector, district magistrate, whatever it may be. They, in his preview, there is a district level committee. So district level committee will monitor how your experiment going, where you are conducting, is there any effect or where to, they will localize to suggest to you this area of the district you need to target your field studies or your experimentation. So this is this study. So again, there are the different committees. Most essential committee as for the researcher major to the our application is fall under the Institute Biosafety Committee. If your application like our application to produce the knocking out of the animals, they have been going through the RCGM. Once is approved, I think to very few applications they will reach to the GSC, and GSC major application if it reach, they will reject or they will consider this really needed or not, reject. If they approve, then need to contact to the state government if you are clinical trial to the respective state. If you target tooth state, you need to take the approval from the tooth state. And again, within the tooth state, then again recommended to which the district you are be targeting your the field trial. This is the all the statutory committees. Now the workflow, how these work if you wanted to take an approval. First, for example, I am the researcher. I wanted to work on, for example, the recombinant expression of the protein, for example, insulin into the cell culture. So I fill the application. There is a different forms are available on the IBKP portal. You need to follow. For animals are different. For medical purposes are different. For infectious diseases are different. And for the crops are different. So you need to fill that required application. After filling of application, you need to submit it to the IBSC. IBSC, every institute they have IBSC, they will see your application, they consider it, and if they feel that this application under their preview, they will approve it. If your application is not under the preview of IBSC, they will recommend it to the RCGM for the approval. So once, for example, your application for the recombinant expression protein in the milk, this application will goes into the RCGM. Again, the RCGM will see your application. If RCGM has an authority to approve it, they will approve it, because for all the field experimental purposes, RCGM has a whole power. They can approve all the application to produce any kind of the genetically modified organism. Organism means animal, plants, crops, anything in the institute level, they will approve it. So it will get an approval. If you have developed anything and your application wanted to do some field trial, then it will go to the GEAC, particularly for the agriculture application. For example, animals and agriculture, it is related to the ICR will again monitor that your field trial experiment, the data need to be shared. What is the data or the recommendations? And like the ICR has been recommended the release of the GE mustard, but it's not been approved yet. So similarly, the ICR will see all any crop varieties or the animal varieties or the animal products has been developed. See, once it has been approved, then there is a final authority to decision make. And there are a couple of, if it's related to the agriculture, again, the Ministry of Environment and Forest will see their impact, long-term impact on environment and forest. If it has been done, it will be approved. Then again, if it is everything done, now it is for the release for the human use, that is for the agriculture production I'm giving. Then the seed, this is the Seed Act. The Seed Act rule is there. The Seed Act, the rule is there. So for the particularly for the crops, it is the Ministry of Agriculture will approve. Now, according to the new guideline for the animal, Department of Animal Husbandry and the Department of Animal Husbandry and Dairying will approve related to the this is what the animal products. Now the safety level, what are the safety level is available? If you talk about the safety level category 1. So this is category 1. This is a category 1. So for the category 1, if you look at the risk group, this is organism, particularly for the microorganisms. This is related to microorganisms that likely to cause the human and plant diseases fall into the category 1, that for require the BSL-1 facility. This is a BSL-1 laminar code or your laboratory should have. Similarly, if you talk, we will discuss about these animals. Okay, so in animal BSL-1 facility, the category 2 means moderate individual risk. So if there is a moderate individual risk, you need to have the BSL-2 facility, and then BSL-3. It will category 3 and above. For example, higher risk, or the 4 example like the COVID, like the coronavirus, which is a high infectivity, that microorganism or that gene will transfect, or that is, all these microorganisms will transfer from one human to another human very rapidly, and there is an intense threat to the human health and rapid transmission. In this category, you need to have the BSL-3 above facility. For example, earlier when the COVID has been started, even for the testing, the very few laboratories in India, in ICR, only the 3 laboratories of the ICR, one is in Bhopal, one is in Hisar, and one is an IVRI, which has a BSL-3 facility. They are allowed to test the COVID testing. But now after this, the data is generated, now it has been allowed for the other laboratories too. Now coming into the genome editing draft, so I will not be going to there because already I have been discussed this. What are these the groups? Now the new groups. This is just I will take 1 or 2 minutes again. This is a group 1. So in group 1, what happens? Single or few base pair edits. Edit means insertion, deletion, lead to the least complexity. If there is a least complexity, then this will fall into the group 1. And to take an approval, this is a new guideline. This is not yet approved. This is a draft proposal. That was I was talking about. If this the draft has been approved, then we will follow a similar guideline to the United States, Brazil, and Australia. So if our animal or your experiment will fall into this will easily approved. For example, in our experiment, which we are working in our laboratory, there's a double muscle mass animal. They are fall into the group 1. Now the second into the group 2 animal category. In this, what is a group 2? Several base pair deletion. Base pair means that is a nucleotide. Base pair in either addition, deletion, and recipe, and that lead to the least, again, the certain degree of complexity. For example, TB resistance cattle. So what happened in TB resistant cattle, if you recollect correctly, that I have shown that NRP1 gene, which is expressed in the macrophages, which enhance their innate immunity to, because the TB bacteria, the mycobacteria has been grown into the macrophages. If you increase the immunity, that will cause the certain degree of complexity, but that will not change any attributes, physiological or the phenotypical attributes more. And these cases will be fall into the group 2. Again, this group 2 animal to get an approval of this kind of the work is easy. And there is a prior knowledge should be there. Prior knowledge means this mechanism has been proved either in laboratory animals or their mechanisms well known. Now coming into the group 3 genome edited category. In group 3, again, this is a similar to the transgenic animal or genetically modified animal, in which the insertion of the foreign DNA or sequence that lead to the higher complexity of the phenotype and genotype. For example, secretion of the human insulin, therapeutic or recombinant human insulin into the domestic animal meat, domestic animal milk. So what happened there is no previous knowledge we have that is the no animal is like this, which is an express the human insulin into the milk. So that will also lead this kind of the changes. They will lead to the new attributes, new phenotypes, also significantly change the new pathological, metabolic pathways. And this kind of the work will fall into the group 3 category. And to have approval of the group 3 category, so even if you have to produce any live animal, your application will go to the RCGM. Now coming into the pathway, again, the G animal, particularly if I was just talking, so the GU animal, particularly for the GE animal, this is IBSC you need to submit, then your application will go to the RCGM and GEC. So if you have RCGM approval, then is okay. You can initiate your experiment. After anything, the final approving authority is the Department of Animal Husbandry and Dairy. So this is a new guideline. Again, I'm stressing this guideline not approved. Don't consider that is 100% approved. It has been draft proposal of the Department of Biotechnology and is available for the public comment. If anybody wanted to suggest anything to improve anything in this proposal, they are welcome. They can suggest anything. Again, there is a different category: Group 1, Group 2, and Group 3. There is a complete assessment of established unintended changes for example, incorporation of the human insulin gene into the animal genome, and it will require the studies for the years, the 5 years, 10 years studies to get an approval of this kind of the animal. But the Group 1 and Group 2 animal studies, it will be approved. I think I was being a little bit running a short of time. I will try to quickly finish. I think a couple of more slides are there. Regulatory, particularly if you talked about the human, suppose somebody wanted to use on the human, suppose somatic cells and the germline cells. So again, there is a restricted use. Yes, if you remember, the restricted use means the regulatory agency, IBS and RCGM, will think your proposal will be useful to the human application or not. If they feel that this proposal need not to be approved, then they will reject. There is the restricted use, which you have been following similar guideline to the United States. Somatic cell, they need to be how molecular characterization need to be performed, their immunogenicity, all the tests need to be done. That is called, suppose the transgenic or genome somatic cells are there, and that's for the human application. Then again, you need to take an approval from, as particularly talking about the human application, the RCGM is a final authority, RCGM or CDSCO, that will authority to approve all these the products or the cell lines or the transgenic cells which has been used for the clinical trial in human. They have been approved. This is about the human application. Now these are some of the guideline drafts. Those are really interested to work on these G technology, G technology. So it contained the DNA safety guideline 1990, 1998, and this is one recent draft guideline which I was been sharing with you. Regulation of guideline for recombinant DNA and biocontaminant. Need to follow this document. I again urge you all to download this document who are really interested, go through it and see in which categories fall. Again, for the field trial, clinical trial, Ministry of the Environment and Forest, the newest guideline is the monitoring or confinement field trial regulation 2008. The guideline for these assessment, safety assessment, it is a DBT, ICMR and ICR is a part of the safety assessment, and the guideline is updated in 2016. Again, the safety assessment for the environment, environmental safety assessment, the G guidelines stakeholder. These are guidelines that are freely available, or these Indian guidelines if you were really wanted to work on the genome edited or transgenic or gene modified microorganisms, crops and animals, then please go through it, because sometimes the lack of information may give us a serious trouble. I know that some of the people, because of their lack of awareness, they have been done the work, and later they have been serious consequence for the career. So as a researcher, our aim was to not to do the science also, but in addition to that, we wanted to follow whatever the guidelines. If you have guideline, if you take the proper guideline approval, then there is nobody can challenge you, or nobody can target you that you have done this kind of work. So first and foremost important, before starting any kind of the gene technology work, take a proper approval from the respective committees. Thank you very much. I hope I could give you some of the information regarding the regulatory agency and what has been allowed for the genetically modified microorganisms, plants, and animals. And particularly, again, urge you to follow the handbook of the IBSC 2017 draft for your research experiment. Thank you. Thank you all.
Thank you very much, sir, for that informative lecture again. And as usual, we have got enlightened with all the new regulations which are existing at the moment. And Dr. Vaskar, if there are a few quick questions, can we take them?
Sure, ma'am. I'm just going to the chat box. Yeah, thank you, Dr. Naresh. It was a very enlightening one, and particularly for the younger researchers who want to pursue their career in this new field.
So I would just like to summarize it a little bit. Sir, your lecture provided us an insight on, production of bovine respiratory disease, that is including tuberculosis and resistance to Mannheimia hemolytica. I think that part was very interesting for us. Production of double muscle mass animals, that is also a very good area. Insertion of human lysozyme genes in buffalo mammary gland, that is again a very interesting area, and developmental gene in buffalo embryos. So that was for the morning talk. And in the evening talk, and the afternoon talk, you gave us an insight in the regulatory guidelines, gene editing, and the draft guidelines has been prepared by DBT. That is a very encouraging news. And all the regulatory frameworks in India, you so elucidly told us, especially about the Indian Biosafety Knowledge Portal. I think most of us will be very eager to go onto the portal and find out what will be the, if we have any queries. So I thank you once again, sir, for your very extensive talk. and a lot of data which you have shown us, and so painstakingly, taking the whole talk. I really appreciate it, sir. Thank you so much.
Thank you.
Yeah, Dr. Naresh, I think there are no questions as if now I could see in the chat box, but I have one from my, which could be in the mind of many of the listeners. How you can break the misbelief that the GMF or GMOs may alter the genomic structure or maybe of the plants, animals, or the humans?
Hello, I am audible.
Dr. Vaskar sir.
Yeah.
Your comment, please.
Hello. Hello. Yeah, Dr. Naresh. It was a very common thing. I think he has lost the track.
Hello, Dr. Vaskar. Should we end the session now only? Or
Hello.
Okay, but he's. I think the presenter is not been seen.
Oh.
Hello.
Because I can't
Hello.
see him on my mobile.
Audible. Hello.
Yeah, Dr.
Yeah,
Naresh.
he's there. He's there.
Yeah.
There was some there's a disturbance in my connection. Can you repeat? Somebody has been asked something, so I could not hear exactly what
Dr.
they are.
Vaskar has asked, I think.
Yeah. It was the same thing. Okay, since there are no questions, only one thing that maybe it's in the mind of the all people, that how can we break the misbelief that GMF or GMOs may alter the genomic structure of maybe the plants, animals, or for that matter for the humans also, because
no, there is a required awareness. Like we have been doing Andhashraddha Nimulan awareness. Similarly, as a scientist, those working, we need to make a massive campaign to take the many misbeliefs about, because we have been eating even in the crops, in the plant, there is always been change in mutation. If the same animal will detect the mutation is always been there, environmental mutation is there. So what we need to have the mass campaign to do these awareness among the people that even if you consume anything, so with DNA, we are consuming DNA, millions of DNA every day. In plants, there is a DNA. In milk, there is a DNA. In milk, there is several cells. Millions of cells are there we are drinking. But from the years and years. So similarly, many people is not aware, common people, but as a scientist, we know. So we need to take a campaign. That's what I would like to share.
Yeah, thank you very much. Yeah, that was a very wonderful talk. We can say the eye-opener for many of the listeners, and particularly it was a very encouraging talk and a new arena we are open for the young researchers or the young scientists who wish to work in this particular field. Thank you, Dr. Naresh, for your very nice and excellent presentation. Ma'am, over to you, madam.
Yeah, thank you once again, sir. As I said, we have already summarized your talk, and I felt that all these areas, that is why I mentioned especially the resistance to bovine respiratory diseases and also the regulatory frameworks which you have mentioned. These all require a lot of insight and research in our country. I think you have really given us good insight. Thank you very much, sir, for your talk. Thank you.
Thank you. Before I left, I must grateful to Dr. Jende for his invitation and the organizing team, Dr. Ingole, Dr. Vilas, and Dr. Sonal. I really appreciate, and my best wishes to your team and your future efforts to continue. Thank you all, and my best wishes to all. Thank you.
Thank you, Dr. Naresh. So participants, coming to the concluding session. Before we start with the concluding session, I would be happy to hear from the participants to have their views about this training session. If any of the participants would like to speak about this training session.
Dr. Ingole?
Yes, madam.
Sir? Yeah, can I just leave if you don't mind?
Yeah.
Yeah, thank you so much, Dr. Ingole, and thank you, Dr. Zende, and the team for having given me this chairmanship of this session.
Thank you,
I thank you all. And I thank Dr. Vaskar for his assistance. Thank you all very much. Thank you, sir.
Oh, any participants, please?
Good evening, sir. Am I
Yeah, good
audible?
Yeah.
Good evening, sir. I am Anamika Majumdar. I am from Guwahati College of Veterinary Science. I am a PG's first year student. So I thank the organizing team. Thank you so much for organizing this training. It is indeed very enlightening and very informative. Thank you so much.
Thanks, Dr. Namika. Any other participant?
Hello, sir.
Yes,
Yes,
Dr. Yes.
Good afternoon to everyone. This is Dr. Gaurav Datta. Currently, I am doing Masters in NDRI Kernel in AGB, Animal Genetics and Breeding. Some of the topics which were of my interest that I needed to inquire in this seminar, I really got very efficient information, especially from Naresh sir. Today's session was very informative about the genome editing in the livestock and obviously the gene cloning and the advances. I think this is the first seminar that I have attended which has raised to his expectations. I really want to thank the organizers. Thank you.
Thanks, Dr. Ramiro. Any other participant? Okay, then coming to the concluding session. Today we are having with us as the guest of honor Dr. A.S. Ranade, Associate Dean, Mumbai Veterinary College, and Chief Guest Dr. A.P. Somkuwa, sir, Director of Instructions and Dean, Faculty of Veterinary Science, MAFSU, Nagpur. To start with the concluding session, I would request Dr. R.J. Zhende, who is the Principal Investigator of this NIAP CAST project under which these training programs are going on, to give his remarks on this training. Dr. Zhende, please.
Okay. Thank you, Sanish. Today we come to the end of this 1 week international training program on the gene cloning advances and applications in veterinary science. Tremendous overwhelming support from all the participants across the country. We have joined more than 48 participants. I got the knowledge about gene cloning. So what are the recent techniques that are used in the farm animals and at the various diagnostic laboratories? So
Dr. Zhende, your internet connectivity.
knowledge and experience, each experience in the field of gene cloning with all the participants. And definitely you will learn so many things, and definitely you can use this knowledge and experience in your day-to-day research activities, what you are doing in your PhD and the faculties are there. So I thank all the participants for for this program events in college. Our beloved associate dean, Dr. is always encouraging the programs for the benefit of the students, those who are pursuing the programs. And Dr. and Dean Dr. Research and Vice Chancellor Professor Sir, he always encouraging for program So with this remark, I hand over to the Dr. Shailesh Ingole. Thank you so much.
Thank you, Dr. Chende. So now I call upon today's guest of honor, Dr. A.S. Ranade, Associate Dean, Mumbai Veterinary College, to give his remarks.
Very good afternoon to everybody. I know we are running behind time. I won't take much of your time. Everybody must be hungry as well. At the onset, I would like to thank the organizers of this program and the people behind it. Professor Colonel A.M. Paturkar, Dr. Somkuar, Dr. Kurkure, Dr. Hare, all other university authorities, they have been encouraging us to organize these type of programs. And of course, our team here at Mumbai, Dr. Jhende, Dr. Ingole, and all others, they have taken efforts to organize programs one after the other. And this was one of them on gene cloning. This was a week-long program, and the response was tremendous, as Dr. Jhende explained. Actually, this particular topic or this particular aspect is relatively new to us. In 1980s, when we were students, nothing of this was available. Everything was Greek and Latin for us. At the most, ELISA or RFLP, these were some of the words which we were knowing in 1980s when the molecular aspects of everything started developing. After that, I believe with the inventions and discoveries one after the other, especially when the computers were introduced in the science, the progress was very faster. However, what I must emphasize over here is that whatever research is being carried out, whatever work is being done, it should be useful to the society. It should have practical importance. It should have practical application. Something to be done which is only of academic interest has no meaning. It's fine for publishing a paper. It's fine for doing a master's or PhD degree. But as long as it doesn't get translated into the day-to-day activities, maybe of the farmers or the owners of the animals or the other activities which the society does in response to the veterinary services or veterinary facilities, unless that is being done, it doesn't carry much of a value. So my sincere request to all the scientists who have attended this particular program is use this knowledge in practical sense to be of use to the society. Whatever field you may be working with, you have to use it for the benefit of the society. Don't just look at it with the academic interest. Like for example, I can quote some of the examples. Just to find the genetic distance between two breeds, you need not use any of the molecular techniques when you can apparently look at the differences between the two breeds morphologically, phenotypically, and you can say this is breed X or this is breed Y. Or you give me anything and I can extract protein from that. That's fine. As long as technology is concerned, that's a great technology. But in country like India, what are you going to do with that protein once you extract it? That's the question. And unless that kind of aspect is related, that utility aspect is related to the work that is carried out by the scientists, it doesn't have much of a relevance. So I earnestly would like to request all the scientists to carry out the activities and the work which is applicable to the society through the science. I could get some time to attend the lectures, and I must take two names. One, of course, my professor, my teacher, when I was a student, Dr. Bhushan Jayarao. Excellent speech or excellent topic he has dealt with. And second is Dr. Kuchipudi. Both of them, they deliberated very nicely, and it was appreciated by one and all. And I also, because by reading the names of the people who are going to speak, I also could attend it for some time, and I was very much happy with that. That doesn't mean that other speakers were not good. They were also very excellent. But these two emphatically I am saying because I attended them, I have witnessed the talk, and I was very happy with them. And from the response of the audience, I can guess that all other topics and all other lectures were also very, very good. I could see right now also there are some 176 plus people who are attending. And as Dr. Jhende said, the registration was some 400 plus. That speaks volumes about the success of the program. And I congratulate Dr. Jhende and his team for such a wonderful, successful program which he has organized. And I wish him all success for his future programs and endeavors as well. As I said, we are running short of time, so I won't extend my talk anymore. Thank you very much for giving me this opportunity to share my thoughts with you as guest of honor, and I wish you all the best for future. Thank you so much.
Hello, can you? Yeah. Thank you, Rna sir. So now I call upon today's chief guest, Dr. A.P. Samkua sir, Director of Instructions and Dean, Faculty of Veterinary Science, MAPSU, to give his concluding remarks.
Good afternoon. Am I audible?
Yes, sir.
Thank you. Yes, today's guest of honor and also Dean, Mumbai Veterinary College, Dr. Jhende PI of this CAST team project, his team, organizer, co-organizers, and their participants. Since morning, actually, I was trying to attend. I was there in and out for these topics. Yes, of course. And as Dr. Ranade expressed his view on similar line, also actually I was trying to attend many of the speakers, and maximum of them I attended. It was so because as we were student that time, this science was just budding. We could not get chance, and thereafter by one or other reason, but I could not restrict myself to know this science. And after going to the lectures, but I personally found that yes, this was a training which was organized in a well manner, and right from gene cloning, its introduction to its pros and cons and each and everything, right from lab to land, I can say, means applicable aspects were there and laboratory aspects were there. In future, what are the scopes? They were also told by some of the speakers, and what are the challenges? They were also told. And today, just now I was listening to Dr. Naresh. He has also expressed in much more and simple way. I must congratulate all the team of Mumbai Veterinary College for organizing this e-training, 1 week e-international training on this gene cloning, its advances in veterinary science. It is required very much, and I'm very happy that maximum of the participants, they were there throughout the training program, and hope that whatever knowledge they gain through this training program, they will apply in their, I can say, their research activities and wherever they are, for whatever purpose they are doing. As it was said by Dr. Ranade sir that yes, it should be applicable. I do agree with that, that it should be applicable, but at the same time, when we get the fruit of the particular thing, so it takes some time. Many of the things are there that we can't see. They may be directly or indirectly applicable. So whatever research the scientists they are carrying out, of course, it will share to some extent to the applicability of these techniques. So lastly, when we can see, as it is said that yes, when we can see some iceberg in the sea, so whatever portion we see, that is only 1/10, but 9 part they are below sea that we can't see. So that means iceberg is much more bigger that actually is there. So in the similar part, I personally feel that yes, whatever research is being carried out, it will be helpful directly or indirectly for betterment of the society, betterment of the health of animal, and thereby health of the human being also for the society. So without taking much more time, it's because we are too late and we are having other assignments also. So lastly, just I would like to congratulate all the participants for completing their e-training program, international training program of 1 week. So I congratulate all of them, and at the same time, I congratulate PI Dr. Jende and his team for selecting and identifying renowned speakers for the training program. They were very, very, very nice, what I can say. And these participants, they will definitely take benefit out of that. So lastly, all of you, please take care of your health and stay safe in this pandemic, which is decreasing day by day. Hope it will decrease to greater extent. Thank you very much. And lastly, thanks for calling me and thank you for your future endeavors. Thank you very much.
Thank you, sir. So now I call upon Dr. Vaidya for vote of thanks.
Yes. Thank you, sir. Good afternoon to you all, respected dignitaries, speakers, chairman, co-chairman of today's session, and participants from different universities from India and other countries for joining to validatory function of one-week international e-training program on gene cloning advances and applications in veterinary sciences. Yes, today is the last day. First, I would like to thank the ICR authority, Dr. R.C. Agrawal sir, National Director, and Dr. Prabhat Kumar sir, National Coordinator, for funding such a nice project, World Map project to our University MAPSU. I also express my heartfelt thanks to our Honorable Vice Chancellor, Dr. Professor Colonel A.M. Patrulkar sir, for his always support, encouragement, and guidance to us. I would like to take this opportunity to thank today's chief guest of validatory function, respected Dr. A.P. Somkar sir, Director of Instruction and Dean, for his valuable speech and always valuable guidance and encouragement to us. Thank you very much, sir. I must thank our today's guest of honor, respected Dr. A.S. Ranade sir, our beloved Associate Dean, Officer, as well as Post Director, for always sparing his valuable time and his speech. He has enlightened us, and always his support, encouragement, and guidance is there for organizing this event. Thank you very much, sir, for your always support and guidance. I must thank the all the university authorities, dean, directors, and all the associate deans of constituent colleges of MAPSU for support and cooperation. I express my thanks to the speakers of different sessions for accepting our invitation in a very short period of time and for sharing the experiences. Especially today's, I am thankful to Dr. Naresh Shelokar, Dr. Kaushik, wonderful and excellent presentations. I would like to thank technical session committee chairman, respected Dr. ASB Maji madam, and co-chairman Dr. V.S. Vaskar sir, for their valuable time, and they have conducted the session successfully in time. And also I am thankful to all the chairman and co-chairmen of departments. I also extend my thanks to the course director and PI, Dr. Jende sir, then organizing secretary, Dr. S.D. Ingole sir, Dr. S.A. Ingde, co-organizing secretary, Dr. R.V. Gaikwad, Dr. Gandagi Patil madam, Dr. Shukla, Dr. Sawane sir, and all the co-PIs who has taken the efforts for organizing this event. I am also thankful to all the project coordinators and co-coordinators. They have taken the efforts. I extend my thanks to all the participants from different universities, institutes across the India and abroad. Because of your participation and support and cooperations, this program meaning, and your suggestions, your questions and answers, definitely that will help us for the improvement of the coming training programs. Thanks to all the professors of our Mumbai Veterinary College and students also for participating in this program. Last but not the least, I appreciate the efforts taken by the NIAC team, all the staff members, students, of MAPSU. I thank all of them for making this event grand success. Thank you one and all. Thank you so much.
Thank you, Dr. Vaidya. Lastly, I would just like to inform the participants that the feedback form will be, the link for the feedback form will be sent on the WhatsApp group of our training program. And you kindly fill that form, and accordingly the certificates will be issued within short time. So I declare this training program over. Thank you all.